Hello, hello. I don't know why this keeps on happening. I don't know why every time I go live, OBS decides to forget which microphone I'm using. Hello, everybody. Welcome to the stream. Welcome, everybody. Hello, Dreamless. Hello, Vietran. Hello, Kingwin. Hello, Ari. Hello, Jacob underscore. Sorry if I pronounced that wrong. Hello, RSY. Hello, Hanifar. Hanifaro. Hello, Nicholas. Hello, Spacraw the Younger. Welcome. Hello, Marlon. Welcome. Hello, Sauce. Hello, Theta. Hello, JV. Hello, Archie. Hello, Leo. Welcome. Hello, Sang. Hello, Neurotic. Hello, Wolfie. All right. Let's get right into it, shall we? Today we are going to be talking about the biology of brain drainage, which I'm very excited about. You can see here already we have some a beautiful image of the brain, and I want to talk to you all about how they managed to take this picture because it's really cool. So I'll just continue saying hello Hoyd, hello Colino, hello Lage Bond, hello Blue Kyogre, welcome, hello Jabu, welcome. So glad you could all make it today. So yeah, this is quite a doozy of a paper. You can already tell by how many authors are listed on here that this paper, cell paper, was certainly a labor of intense love. And today we're going to go over what that science entails. So let's get started. So it's actually been a while since I last did a journal club. So I wanted to make sure that for you all out there that we all kind of understand the format that we're going to be doing today. So we are going to be doing a journal club. It's a modified journal club. But what is journal club? Why do I do this? Okay. So in an academic lab, researchers will all agree to read a paper while one of them presents on it. So this is how this works. This is because it's actually really hard to fully understand the nuances of a paper alone, right? So you can read a paper on your own, but because we are operating at sort of the highest levels of human thought, it usually takes more than one person to fully appreciate the nuances of a paper. And I'm not claiming to be able to do that for you. I'm just one guy, right? Since I'm no longer in academia, I don't actually have the environment that pushes me to stay sharp. So, part of the reason why I do these is because I want to force myself to feel the discomfort of not knowing, right? Brady says researchers will all agree to read a paper, all agree to skim a paper. Shh, you're not supposed to tell that to the young students, Brady. Okay, you should read the paper. Okay, you shouldn't just skim it. You should actually read it. You should. But yeah, most researchers will just skim the paper. The person presenting will be the one who usually reads it in depth. But if you want to push your understanding of things, you should genuinely read the papers as well. Okay. So yeah, because of my new job, which is to do YouTube, I don't actually read very deeply anymore for these videos, right? I'll read the abstract, I'll skim over the paper, just as Brady says most academics do. But for a journal club, I actually really do take a lot of time on one paper. It takes me about, a couple of hours to make it through one paper for a journal club. Whereas for a video, I'm usually not thinking very critically. I'm usually reading to understand and not reading to fully deeply interpret, right? So there's a difference in how I treat reading for stream and reading for a video. The goal here is not to be an expert in this paper, but to push yourself very deep into a topic and see what you can learn. So as I've mentioned repeatedly, that poll I did about asking what the biology training of my audience was really useful because I now know that a lot of you don't have deeper biology knowledge. And so that's fine. You're going to be right at home here, but you are going to be uncomfortable. I need you to know that right now, I'm gonna need you to know that you will feel lost, but that's actually not a bad thing, right? This is difficult, right? It's still fun to be around even if you don't have a lot of biology knowledge, because I've kind of blended this journal club with a lecture to try to ease you into these higher levels of thought, right? And yeah, if you're having a hard time, that's actually good, right? You're using your brain, and that is going to feel uncomfortable. It's going to feel difficult, but it's still going to be pretty fun. At least your animation takes less time on the journal club. That is true. Note that this is a modified journal club, a hybrid journal club, and a teaching tool. So if you are a young student, do not do what I am doing here. I am going way more in depth, and also I'm asking you questions you would typically not ask an audience questions in a journal club. So don't imitate the way I do this in your labs. This is, a modified journal club for content and for education. So before we begin properly with the subject of brain drainage, I'm going to take you on a dumb rabbit hole that I went down to try to make the introduction to this video. So I had a whole opening that I thought that I was going to do to introduce this topic of brain drainage, but it was absolutely ruined by how complicated the brain ended up being. Go figure, it's the brain. It's quite a complicated organ. And even though I studied the brain in graduate school, I'm still learning a lot whenever I force myself to do some of these streams. So the original introduction to this journal club looked a little something like this. I was going to start off by opining on how important garbage collection was in the brain. Going to talk about why it is important that garbage is collected from the brain, and the reason why it's important is because the brain is extremely metabolically active. Your brain consumes a lot of calories, a lot of calories. But the problem here is that metabolism in the brain is actually really complicated, and sometimes for no good reason in terms of the language used. Thank you so much for the gift, Pathogenics. Thank you so much. That's awesome. That's so fun. Thank you. So what do I mean by that? So the brain has a bit of a metabolic paradox here. Okay. And what do I mean by that? So how many of you in chat right now have heard that the brain primarily uses glycolysis to fuel its function? This might be a fact that maybe not a lot of people know, but I'm just gonna poll the audience. How many of you have heard of this before? That the brain is sugar hungry, that it eats sugar like nothing else in the body. It craves sugar and breaks it down with glycolysis. How many of you have heard of this before? I probably have heard, but not remember. Yeah, it's one of those fun biology facts that is not necessarily that fun if you're not a biologist. But yeah, does this include the part of the brain that is not neuron? I'll say generally yes. I have not me. Feel like I have. I've heard. Yeah, so we have a good mix of different exposures to this fact, right? Not the medical interpretation, but just sugar makes brain work. Yeah, the brain needs sugar. Yeah. I feel like I've heard this before, but cannot recall where or when since it slipped. Yep. I've heard of it. The thing is, and this is something that confused me as I was reading this, is that glycolysis is classically anaerobic, meaning it does not require oxygen, which, I've heard of this before too. The brain uses glycolysis, but it didn't really mesh well in my brain to be like, but this contradicts something, doesn't it? And before I get into the contradiction or the paradox, the fact that the brain uses sugar is actually pretty understandable, right? Fats and lipids are too big to cross the blood-brain barrier, so alternate pathways of energy consumption are kind of off limits to the brain because of that. So sugar is really one of the only options the brain actually has. But the paradox comes in when you learn that the brain doesn't actually seem to be that good at fully oxidizing that sugar, right? Which is an aerobic process, requires oxygen, right? We've heard of mitochondria, right? Mitochondria, powerhouse of a cell, famously dependent on oxygen for the full breakdown of glucose to carbon dioxide and water and some odd 36 ATP. But the brain, you will read online that it is primarily dependent on glycolysis, which does not finish the job, which is kind of bizarre, right? Kind of bizarre. However, that doesn't seem exactly to square with the knowledge that the brain needs oxygen to function. So we have a bit of a weird kind of overlap in knowledge that I didn't really confront before, but there is a solution to all of this. It's just when you first read about metabolism in the brain, it is kind of confusing. So I'm making you walk through my shoes, to see some of that confusion, right? Marlon asks, does this result in a lactic acid buildup? That is interesting, right? That is interesting. 2 ATP versus 32 ATP is my favorite biology fact. I like to bring it at parties. You must be very fun at parties, Brady. So this has always been a weird contradiction, right? The brain uses glycolysis, is what you will read, but glycolysis, A, isn't that efficient on its own. You need to fully break it down to, as Brady said, break out that 32 ATP. So, this contradiction really bothered me, and I went down a rabbit hole to try to resolve this. So the brain actually uses a special form of glycolysis called aerobic glycolysis in the presence of oxygen, which is interesting. So despite the availability of oxygen, the brain does not fully commit to oxidative phosphorylation, which is what happens when you have mitochondria fully breaking down sugar. So despite there being a lot of oxygen around, the typical mitochondrial pathway to use oxygen to generate ATP is still underutilized, but it's still used. Okay, this is why this is so confusing. People online will say that the brain relies on glycolysis, which is true. It uses glycolysis a lot, but that doesn't mean that it completely ignores the mitochondria bit. It's just that these are context-sensitive facts. So instead, the brain goes halfway between glycolysis and oxidative phosphorylation in a pathway called aerobic glycolysis, whose final metabolic product is lactate instead of pyruvate, right? So pyruvate is the thing that gets fed into the mitochondria typically to give you the normal energy yield that you would expect. So this actually interestingly means that the brain shares some metabolic similarities to immune cells like neutrophils and cancer, right? These are all cells that can produce energy, a lot of energy, even in the absence of oxygen. But, there's this, again, it's confusing. It doesn't fully make sense, but I promise I will get there. Thank you so much, Pathogenic, for being a member. Oh, for the super chat. That's a super chat. Sorry. Last time I donated was the day before my hematology and immunology exam. Get results on Monday. Good luck. Your stuff really helped me. Wish me luck. Good luck. Good luck. Good luck. Yeah. So in this abstract, in this paper, you can say, why engage in inefficient ATP production by glycolysis when energy demand is highest and oxygen is plentiful? It's true. It's an interesting, very bizarre metabolic conundrum. And as Brady had pointed out, the virgin glycolysis, 2 ATP generated per glucose, 2 NADH generated per glucose, not a whole lot of energy per molecule of sugar. And then we have the Chad bespoke aerobic respiration where you get, the numbers vary because this is not perfect every time. The efficiency isn't exactly 32 ATP. Some say 36, some say 38. But, fully breaking down that glucose is going to get you a lot more ATP. So why does the brain seemingly just rely on glycolysis? And so the reason is complicated, and I swear I didn't use AI to get. I was tempted. I'm gonna be real, right? I'm gonna be real because I think it harms society to ignore how convenient AI can feel. But at the same time, I want you to know that instead of just asking AI, I just spent, I think, maybe a full hour trying to get to the bottom of this stupid thing. So why does the most energy-hungry organ in the body waste 30 ATP per glucose? Is the brain stupid? It's really hard to get to the answer to this. I just wanted to express my frustration here. It's really tempting to get the answer easily, but, it is valuable to sit in the discomfort of not knowing and just power your way through. The AI will lie, best not to ask. Exactly. Like the AI isn't even very accurate all the time. But I think if we want to set examples for people who want to wean themselves off of AI usage, to acknowledge that it is so tempting is important. And then walk through the process by which you can talk yourself down from using an AI. Whenever I feel like oh, I just want the answer, like I can, that's a symptom of like the brain kind of like giving up, and it's important to not give up, So how does this actually happen? So one contested hypothesis is the lactate shuttle hypothesis, and I say contested because this is still widely debated, even the big 2026. So what this says is that yes, a lot of the brain's activity is driven by glycolysis. That's because astroglia are the ones that are doing the glycolysis. They're eating up the sugar and producing energy really, really quickly. And then they are passing off that lactate to the neuron. So lactate is the end product of aerobic glycolysis, and the neuron can actually use that lactate as energy on demand, right? So it stores this lactate, and then it can use that lactate, break it down fully for a lot of ATP through oxidative phosphorylation, which contradicts what I saw earlier when I was researching this stupid topic, because, as we saw in that earlier abstract, people were saying that, the brain relies on glycolysis. So why, this seems like a contradiction. So even in 2025, this remains kind of controversial. It's a lactate shuttle hypothesis. It's not entirely clear that neurons get lactate from astroglia, and whether or not neurons are burning it for energy exclusively or even in significant amounts, it's tough to tell. So here's the mess of this stupid topic, right? So the brain relies on glycolysis. The brain doesn't fully break down glucose through oxidative phosphorylation, but the brain uses aerobic glycolysis to make lactate and then fully breaks down lactate, which is a glucose byproduct, which means it does use oxidative phosphorylation. So how does this contradiction get resolved, right? So I think SciComm needs to be a lot more careful with throwing out this line of the brain uses glycolysis primarily, because that is a gross oversimplification. And what this ends up saying is what the research, as far as I know, what the research ends up saying is the fact that when you are resting and awake, glucose is in fact the primary metabolite that or the primary fuel your brain uses, right? So your brain uses that glucose. Sorry, this should be sorry, this is the reverse. So when you are resting and awake, glucose is fully broken down to CO2, water, and 36 ATP, right? So the brain is just like any other organ and using that glucose just like any other organ would, fully breaking it down for the maximum amount of ATP. However, when neurons are activated, when they are very active, when you are doing stuff, when the brain is doing stuff, then you go halfway. Then those astroglia are crunching down on that glucose, producing lactate. And then what this lactate does is not entirely clear, but then this lactate gets broken down as needed, which generates a lot of metabolic byproducts, as well as carbon dioxide, water, and 36 ATP. So the claim that the brain primarily uses glucose in the form of glycolysis is true for the active awake brain. And there are some hypotheses that that's because the brain, that glycolysis is very fast. And then the lactate kind of builds up, and then there's a slower phase where neurons might be able to use that lactate for the full yield later. So it took me about an hour to answer this question, because whenever you Google, metabolism in the brain, you get this very simple top line summary: the brain uses glycolysis. When it's like, yeah, a lot of things use glycolysis. Does it use oxidative phosphorylation as well? And the answer is it depends on whether or not you have an active neuron or an inactive neuron, or there's high levels of neuron activity or low levels of neuron activity. Anyway, does this have to do with today's journal club? No, it doesn't have much to do with today's journal club. I just wanted to share this because I spent time on it, so I might as well make use of it. Might as well make use of it. Okay. Piles and piles upon AI generated web page SEO glop. Yeah. That's the thing too, is that if you, I love DuckDuckGo because you can remove the AI search function on it, right? You can just remove it, but I don't think Google allows you to do that. And right now, Google still beats DuckDuckGo in terms of search results that are relevant for me. So when I say that, like, the tempting, because it shows up on every Google search, the AI thing, it gets very tempting to go in that little search box because it's right there. It's so tempting to just give up that mental strife. But just having the fortitude to ignore it is not that easy. And I think if we acknowledge that it's not that easy, more people will feel heard, understood, and then be able to walk themselves out. I think that AI is here. It's here to stay. It's terrible. But in order to get people to stop using it, we can't ignore how tempting it is. How tempting it is. I believe there's an ad blocker or some add-on to block it. Yeah, maybe I'll look into that, because it sucks being tempted with the AI search tool. It really does. Google Scholar doesn't have AI search, does it? So Google Scholar, I do also use primarily, but when it comes to a more basic question, I usually go with Google Search instead of Google Scholar, because Google Scholar takes a bit more time, and also you get led down a lot of false rabbit holes instead. Google Search is better for like a general understanding, and then you go to Google Scholar to go in depth, right? Because if I don't understand the basics of brain metabolism, the papers themselves are going to be very, very difficult to read. So it's quite difficult. I found Kagi to be much better than either DuckDuckGo or Google. Well, I'll look into it because I don't want to be staring at the stupid AI search tool every single time. Is DuckDuckGo that terrible? I think it's perfectly acceptable. For research, for me, I have found that I often get fewer relevant results than Google does. Yeah. You're a YouTuber, you know how Google loves to make addictive algorithms. You know what games you have to play and other YouTubers have to play. Yeah, like, and we are being played, right? We didn't really consent to the presence of all this AI being shoved in every aspect of our being, but it would be good to acknowledge that it feels useful. Again, I use the word feel very carefully. It might not be useful, but it is tempting to do so. So instead of just like claiming that I'm never tempted to use AI, I think it's better to acknowledge that it is there, it's being pushed, but it takes an active amount of resistance to stop using it, right? Or to not use it. Yeah. Okay. So all of this to say, the brain is a stupid, complicated organ, and I really do enjoy it, even though it is really complicated. But being so metabolically active means that it's making a lot of stuff, right? And just as a byproduct of making stuff, it's also making a lot of junk, right? A lot of junk. So exosomes, degraded peptides, excess neurotransmitter, carbon dioxide, potassium ions, various sugar metabolites. There's a lot of stuff that the brain makes that is needing to be cleared after it has served its original purpose, right? Anton Petrov had a video recently. Research on mitochondria provides answers as to why we have to sleep. Could the brain use glycolysis to prevent over-reliance on mitochondria? It's an interesting question. I think I actually have a previous journal club on maybe the same paper that Anton Petrov is covering. It was a long time ago, so I don't quite remember the details. But maybe. But maybe. Okay, so for neurodegenerative disease, we must consider two very relevant byproducts, right? Amyloid beta and tau. So how many of you guys have heard of amyloid beta and tau before in chat? Are these familiar proteins to you? And to get a sense of like the exposure to this idea, let me start a poll. Have you heard of amyloid beta or tau? So this will give me an idea of like what our working knowledge of neurodegeneration is. Working knowledge. Just heard of it. I don't care if you actually know it, just have you heard of it? First time hearing, yes, that's scandal. Oh, we'll talk about the scandal. We'll talk about the scandal. Vaguely, mostly in medical heads, oh, that's interesting. They come up a lot in Alzheimer's research. Yeah. Yes, but I have no idea what makes them and why they exist. Interesting. Okay. Cool. All right. Okay, interesting. All right. So this is really helpful for me because as, you know, someone who has a lot of molecular biology education, I sometimes forget whether or not certain things in our field are famous or not. So I'll go ahead and end the poll right now because we're about half of the current viewers have answered. So let me just cut it off there. And we can actually see that we're about 50-50, which is very interesting. To me, as someone who has been very interested in neurodegeneration since I was, a middle schooler, I am happy to be the one to introduce you to these two proteins. Okay, so amyloid beta, we'll start with this, is a major suspect in Alzheimer's disease. So up here we can see in this kind of brown staining, this is staining for the amyloid beta protein. And these darker regions, these are the nuclei of neurons, right? So we can actually see amyloid beta in a cross-section of the brain, a sample of the brain, build up in these neurons, and this can lead to cytotoxicity. Here's the protein alone modeled in protein data bank, so a short protein. So what is amyloid beta? So this is when this protein in the bottom right here, when it builds up in aggregates, when it builds up in plaques, it starts binding to each other, gets sticky, gets difficult to break down, builds up in neurons and around neurons. This is what some people believe to be a causative agent in the dementia-causing disease, Alzheimer's disease, right? And so the only way you can make a definitive diagnosis of Alzheimer's is through a post-mortem finding of amyloid beta plaques, right? So, you know, these days you can get a lot more confidence as to whether or not a patient has Alzheimer's through biomarkers and all those other fancy tools. But the only way to know with 100% certainty whether or not a patient had Alzheimer's is finding these plaques. So no matter what you think about Alzheimer's, I'm talking to the biology heads in chat, no matter what you think about Alzheimer's, you have to acknowledge that amyloid beta is a part of the story. What part of the story, that's still being worked out. So this amyloid beta protein in Alzheimer's disease inappropriately aggregates in neurons, right? It doesn't stay soluble, it doesn't stay happily surrounded with water molecules. They group together in a prion-like fashion. So this is hypothesized to cause neurodegeneration in Alzheimer's patients. So this hypothesis has become more controversial as time moves on, as some of you chatters have already pointed out, and the controversy can be summed by the following question. So, is it correlation or causation? I see Marlon is bringing that up as well, whether it's a causative agent or simply a downstream symptom of an upstream failure. That is kind of the crux of this problem. What's the difference between dementia and Alzheimer's? So dementia is a general symptom, and Alzheimer's is a specific disease. So Alzheimer's has a cause which can lead to dementia. Many different diseases can lead to dementia. Dementia is the general manifestation, the physical manifestation, the phenotype, the expression, the symptom of what a patient is experiencing and shows. Okay, so clearing plaques in Alzheimer's patients provides very little clinical benefit. This has been known for quite some time. But personally, I don't think the amyloid hypothesis should be written off entirely, but other options are looking more and more tempting by the day. I think they're doing something, just not the whole story behind Alzheimer's disease. Okay. So why do I phrase it like this? Well, as someone had mentioned earlier in chat, there was some controversy where a famous Alzheimer's researcher was found faking data, which kind of shook up the whole amyloid hypothesis, because that researcher who is manipulating data was an amyloid hypothesis enjoyer. But just because there are a couple of bad apples, I don't think we should throw the whole thing away. We just have to be a lot more careful about how we think about the role of this protein in this disease. And't get into kind of the nuances of this towards the end of the presentation. How does amyloid beta get made? So amyloid beta comes from amyloid precursor protein. I don't get into this in this presentation, but the amyloid precursor protein is a protein, and then that protein gets cut up through proteases, and if it gets cut in the wrong place, it can form a fragment that is very likely to build up as plaques. How do people try to fake a paper that gets reproduced? I mean, humanity is on a spectrum, put it that way. Okay, so a lot of science drama surrounding the falsification of data, which is sad. Again, I think it's perfectly fine to believe that the amyloid protein is doing something. I think it's pretty extreme to think that it's not doing anything in Alzheimer's disease. I think if you go that far, I think maybe you should rein it back in a little bit. But I think I can agree that maybe this is not the star player in Alzheimer's disease. I think I do agree with that. Okay, so what does amyloid beta actually do? I do go into this. So amyloid precursor protein, this is the protein that is made at first, and then it can go through one of two different pathways: the non-amyloidogenic pathway, which is kind of, you can think of it as like the healthy pathway, and then once it gets cut up by the healthy version of this pathway, it has anti-inflammatory properties, neuroprotective things, and it is negatively regulated. So the more of this non-amyloidogenic pathway gets made, it'll negatively feed back onto itself and stop itself from being made. So it's carefully controlled. It's doing nice things for our neurons. However, when this protein is being inappropriately cut up through the amyloidogenic pathway, then you get this amyloid beta production, then you get plaque formation, then you get this toxic phenotype, and this can actually lead to tau tangles, which I will talk about very soon. Right. So interestingly, non-plaque amyloid beta has neuroprotective effects. This is actually a finding that is more recent. I remember when I was in high school, people just had like no idea what this protein did, and now after a decade or two, now we're starting to understand that actually this protein does actually do some pretty useful stuff, and this effect is actually greater in younger animals, perhaps adding another layer of nuance because we know Alzheimer's disease is a disease of old age as well. Okay. So what about tau? So amyloid beta production, the plaque, can lead to a catastrophe on another protein as well, the tau protein. So the tau protein, the function is a lot better understood. It is a microtubule stabilizing protein. So you can see in this figure that tau protein binds to the microtubule, helps stabilize it in neurons, which is really important because neurons are microtubule enjoyers, right? The body of the neuron is made the axon is made of microtubules. A lot of it is made of microtubules. And this is important because neurons are very long and it needs to transport cargo from one end to the other a lot of the time through motor proteins like kinesin and dynein. So microtubules are really important for the neuron. And the problem with tau that is inappropriately phosphorylated, so it's had too many chemical modifications attached onto it, is that this can destabilize those microtubules and lead to diseased and dying neurons. It's really bad. This hyperphosphorylated tau can actually convert healthy tau into the diseased state as well, acting kind of like a prion. So that's bad, right? You get these tau tangles, nasty aggregates of this protein. So is tau a prion? Well, if you compare tau bundles to prion bundles, they certainly do look very similar. It's okay to say that tau is prion-like. Whether or not you can call tau a prion, language isn't entirely consistent, but I think you're very safe in calling tau prion-like. Yeah. So, in neurodegenerative disease, garbage collection is often impaired, right? Often impaired. So understanding how the brain clears this garbage, especially amyloid beta and tau, as well as what changes happen during neurodegeneration, could hold the future for treating these diseases, right? So if we understand how the brain gets rid of this garbage, maybe one day we can improve the way the brain is getting rid of the garbage and potentially prevent some of these diseases from happening. Okay. So here's the problem with the current state of understanding how the brain drains, is that a lot of brain drainage maps rely on the injection of a tracer directly into the brain, right? So, and by the way, I am reading chat. If I don't address your question, the simple response is I might not know the answer to your question entirely, right? So Seed asks, sleep is where most garbage collection happens, right? I think that is true, but I haven't read about sleep in a while, so I can't confidently say that I know that to be a fact, although I imagine that it to be true, right? So if I don't get to your question, it's probably because I don't fully know the answer with 100% certainty, right? I think I'll only address a question if I'm very, very certain I know the answer. So just for everybody else who's asking great questions, there are a lot that I can't answer because I'm not an expert in this particular field. Yeah. tracers some kind of colorant, not colorant, so they're usually fluorescent, right? So you inject this dye and then you shine a light on them and then they'll give off a glow, and that's how you can trace the brain. So I want you to be the scientist. Why might it be kind of problematic getting a map of the brain's drainage system by injecting a dye into the brain? Why might this be kind of problematic, chat? Go ahead and give me some hypotheses. So Vietran says tracer connecting to other proteins. I'm guessing you mean binding, or binding to the blood-brain barrier. That's interesting. So yeah, you might be disrupting natural functions of other proteins if that tracer, whatever it is, interferes with normal function. I will say typically these fluorescent tracers don't typically affect physiological function. They've usually been tested pretty extensively to make sure that that isn't true. But it's a good hypothesis. Definitely a good hypothesis. The insult of the injection itself might mess up the drainage system. Yeah, interesting, Ari. Very interesting. Yeah, maybe the fact that the injection itself is kind of a violent thing to do, that could mess up understanding how the brain would naturally drain. Yeah, exactly. Do different qualities of the molecule influence how the drainage system transports it? Okay, Marlon, you're going to have to keep that in mind because we will get to that much later into the paper, but hold on to that idea. Different type of trash requires different clearing mechanisms, so a single type of tracer is not enough to fully illustrate. Also an excellent point, similar to Marlon's. I'll get into that later into the paper. And what you're getting at is a bit of a fundamental question with studying the brain drainage system. All excellent ideas. Changing pressure. Yeah, when you're injecting something, you are inserting a bit of pressure. Excellent hypotheses. Excellent hypotheses. Injecting mass disrupts the flow of mass already in the brain. Yeah. Putting more garbage in the garbage disposal. Also an excellent point. It creates new channels. We will see if it does. That's interesting. All really, really good. All really fantastic hypotheses. And I just want to emphasize, you guys are right. And you guys didn't have to be career biologists to figure it out, right? There's a problem with injecting dye into the brain. So here are two answers that I came up with, right? So there are other great responses in chat, but I'm just going to give you the two that I thought of. So the first one is near and dear to my heart because I was a brain inflammation researcher back in the day. So the first answer that I came up with was inflammation. The very act of injection is an insult to the brain, right? You penetrate certain layers of tissue that is going to cause an immune response, and we already know that immune responses can change vasculature, right? So we'll see later on in this paper, but when the brain is attacked or infected with a material that is indicative of an infection, the brain's blood-brain barrier can often loosen. So adding any additional inflammation to the brain is going to cause some funny things to be happening. And two, and this is the explanation that the researchers of this paper primarily give, so if you answered this, congratulations, pressure from the filled syringe can push fluid into areas that they might not naturally drain towards. So all of our understanding of the brain's drainage maps might be maybe not wrong, but maybe not totally correct either. It is very, very interesting. So here is another You Be the Scientist moment. Can you think of a general strategy to label the brain's drainage system without the need to inject a label? If you read the paper, please don't answer. This is just a fun bit of magic that I get to experience with my audience here. So instead of injecting dye, what might you want to do instead? If you can't, or if you should not inject dye, what could you do instead? MRI or CT scans? The problem with CT is that by definition, that is, I believe, a phase contrast. But I don't think, but it's not injected. You're right. Okay, so MRI or CT, I think those are good hypotheses. However, I don't believe they have high enough resolution to resolve individual vessels, which is what you need for brain drainage. So definitely interesting, but it won't actually give you the right amount of resolution. You can't actually see the drainage if you do it that way. But, good guesses, I think. Good guesses. Does it have to be a human brain? No, we're going to be working with mice in this paper. Yeah. Create radioactive atoms in situ. So radio labeling, that is interesting, but how are you going to get it to only label the drainage system? Yeah, that's an interesting problem. Interesting problem. Can you look for an existing convenient garbage protein? Also an interesting hypothesis. You might hold on to that idea for what they do later in the paper. Is it possible to image the proteins? It is possible to image the proteins. It is possible, and they'll actually do that later in the paper, but not immediately. I actually did see the correct answer earlier in chat. Much earlier in chat. Use a virus to add a tagging epitope to certain commonly expressed proteins. Also interesting. It's still probably prompt inflammation. Yeah, that is an issue, right? That is an issue. Viruses, no matter how safe they are still viruses. There's still going to be some amount of inflammation, which is going to be a little fishy. Fluorescently tag. Ooh, okay. So maybe we have a biologist in chat here. Fluorescently tag a protein. That is interesting. Who said that? That chat is very active today, which I'm very, very happy with. But it does make it a bit harder to track. A secreted fluorescent protein produced in the PC of mice or other test subjects. Also interesting. Create transgenic mice model that expresses GFP in the brain. Oh, interesting. All very interesting questions, hypotheses. Yeah, so chat, you're getting it. Genetically modified organism that generates the dye inside the brain naturally. You're getting to their strategy. Awesome. Okay, awesome. You guys are doing great. So that is their answer. So how about a transgenic mouse? So I'm going to need to explain this word transgenic. So a transgenic mouse is any mouse that has been genetically modified. The term transgenic means that it acquired a gene in trans, meaning outside of their body, as opposed to cis, which is inside their body. So their strategy here is going to be to make a genetically modified mouse that makes a green fluorescent dye within its neurons. Sounds woke. Brady, I know... You get me. You know exactly why I had to define this. Sounds woke. Yeah, it's super woke. You know exactly the kind of Congress people I needed to make this clear for. Okay, so I'm gonna pause here, chat. Does this make sense? Okay, does their approach make sense, right? We know, or they're arguing that injecting a dye into the brain is bad because of the pressure that it introduces, which could cause the drainage system to not be accurate. So instead, what they're going to do is they're going to genetically modify a mouse to make green neurons, to make neurons that spit out a green protein to check where that protein drains. Does that make sense? They're turning the mice trans. God bless America. Let me just say that. Yeah, okay. So it looks like okay, so Ari says, will the dye be recognized as garbage? That's the question, isn't it? So maybe we'll talk about this now because you guys are expressing skepticism, which is appropriate, right? So how do we make sure that the brain's drainage system is very natural, right? Very natural. And I think the answer is you can never be that sure. They have an interesting experiment that gets around this later on, but most things that you do to track the brain's drainage system is going to affect it somehow. And I think the best you can do is try to find the way that minimizes this impact, And I think there's going to be an experiment later on that they do, which is going to be really, really useful. But even that experiment isn't going to be perfect. So I'll say that your skepticism would be like, well, isn't this just adding more junk? Yes. We're just kind of hoping the junk is representative of the average protein, and we'll get into how good that argument is going to be later on in the paper. But those are excellent skepticisms that you guys are expressing. Great. Okay. So let's continue. So let's learn a little bit about how they transed the mice. So this is going to be difficult. So if you don't have a biology education, and it's going to be normal that you feel lost here, but I'm going to try to encourage you to try to stick around because this is really, really cool. So what we're looking at here is a mouse, a line of mice called the ZSG transgenic. And if you do !ZSG, you can learn what ZSG is. But basically ZSG, ZS green, is the green fluorescent protein that is going to be tracing our neurons. And that is going to be put at the end of this lock, stop, lock sequence, which is a barrier. Okay. So on its own, this mouse doesn't do anything special. This doesn't do anything special. It's just a normal mouse, all things considered. It just has a gene that could potentially make a green protein, but it's not being allowed to because of this stop sequence. Okay. So the gene for this fluorescent protein is ZS green, but it can't be produced because of the stop preceding it. What's the difference between ZS green and eGFP? I think eGFP might be too big for this experiment, so they went with ZS green. I think ZS green is smaller, but yeah, usually they would start with GFP, but I think GFP is just too dang big, and I don't think cells spit out GFP. Okay. So this mouse was engineered to only make green fluorescent ZS green when it gets a certain signal, right? So by default, there's no signal, right? By default, this mouse lives happily, but once it gets the signal, the stop sequence gets cut out of its genome, and then it can make ZS green. So what is that signal? When this lock, stop, lock sequence is removed, then the ZS green can be produced. Okay, so any molecular biologists in chat who know the answer here. So what is the signal that cuts the lock, stop, locks out of the genome? Again, if you are not a biologist, there's like 0 chance that you get this correct. But for my biology students out there, what is the signal that cuts lock, stop, locks out? Yeah, all right. So we got some biologists here. Cre recombinase, that is the correct answer. So what you can do to cut out this DNA, the lock, stop, lock sequence, is to introduce an enzyme called Cre that acts as molecular signals to scissors to cut the sequence out. So you can do this in a couple of different ways. You can do it with mating, so you get a mouse that is making Cre recombinase, or in this case, you use a virus, in this case, AAV, adeno-associated virus. So you take a virus containing synapsin Cre. I'll explain what this means in just a little bit, but there's a virus in here that contains the Cre recombinase, and it is only made when it is only going to be made in neurons. And why that is because it is attached, stapled to the synapsin promoter. So now synapsin is a neuron-specific promoter. So if you inject this AAV virus into the eye, or the not exactly the eye itself, but the vessels around the eye, you can push in that virus, which will get diffused into the brain. So instead of adding pressure by injecting directly into the brain, you're actually injecting around the eye, which won't produce that puff of pressure into the brain. That virus gets carried into the brain, containing the molecular signals, the molecular scissors. So when you introduce that virus, this is the virus sequence, right? So the synapsin promoter, this makes sure that Cre is only made in the neurons. When that crosses with the trans mice, you put in those Cre molecular scissors, and then you cut out that lock signal, and now the neurons can make green protein. So wasn't the point to not inject anything? So this is injection in a different region, and this is the solution that the authors made in order to only get the mouse to make green protein in the neuron specifically, right? So that's their solution. Otherwise, if you just have a mouse that glows green, then everything will be labeled green. The Cre lock system, if there's one thing that all of my audience can take away, is that when you want specific time and location specific expression of a particular protein, you can use a Cre lock system. In your mouse, you have the locks with the stop, and then you introduce from an outside source, whether it be another mouse or whether it be a virus, the Cre enzyme that is controlled by a specific promoter, in our case it's synapsin, to cut out the stop signal. So if you didn't quite understand that, that's totally okay. This is higher level molecular biology. This is cutting edge stuff. Cutting. But basically, they use a virus to make the scissors that enables this green protein to be made in the neuron. Very interesting stuff. Very interesting stuff. Okay. Tuning the system on briefly with a virus, while clever, doesn't tell you the system continues to work correctly necessarily. That is the correct skepticism to have, but you should also balance that skepticism with acknowledging that not every experiment will be perfect. So while you can keep that skepticism in mind, you should keep in mind whether or not that affects your interpretation of the results down the road. And it's certainly true. I even said that earlier on, any inflammation is going to cause issues, which is true. But I think it would be difficult to find solutions that don't involve at least a little bit of inflammation, although you might be able to get away with some fancier technology these days. Maybe an intraperitoneal injection, but then you get a lot of loss of the AAV, and then maybe systemic issues happen, right? So the skepticism of, yeah, modifying the system might not make it physiological is true and good. But if you want to be a good scientist, you have to balance that with acknowledging that any modification you do is going to affect the system. And you should be thinking about how to minimize those effects. Yeah. Why not use a promoter directly? So they wanted to make sure that they only turned on this gene at a specific time in developmental time. So you could argue that if you just made these mice, pop out of the mom already making green fluorescent protein, that's going to affect their neurodevelopmental biology. And so the way they tried to balance, again, because everything's a modification, the way they tried to balance the effect of making it since birth is through this Kree-lock system, right? Because with Kree-locks, you can control time and you can control space. So they waited, I think, 4 months, I think. And I don't quite remember from the paper. I don't have it here. I think they waited 4 months and then they injected. So we have a fully developed brain. So you don't end up with weird artifacts from forcing a baby mouse to make an extra protein in high quantities. So yeah, they did think about the physiological constructs here. It's difficult to get it perfect. It's a good question, though. Do they make it for the rest of their life? Yes, they do. They make it for the rest of their life, which is going to be relatively short because they're going to be dissected and stuff for these experiments. Yeah. Okay. Is the ZS green produced only in neurons or all cells in the brain? Because the Cree recombinase, because the molecular scissors are only made by using a synapsin promoter, these are neuron specific. Right, the synapsin promoter is only active in neurons. So that's why this Cree scissor only gets made in neurons, which means you only get this cut out in neurons. Yeah. So I just want to back, because I'm seeing a lot of pushback from chat, which is fun and valid. But again, we just got to call it somewhere. So modifications are going to affect the system in some way, Okay. All right, cool. Let's continue. So not only will neurons make ZS green, they also secrete the protein, so ZS green is secretable. So you can see that after 14 days after this AAV scissor has been injected into the mouse brain, that the neurons will glow green. And not only that, the neurons will also spit this green protein out, which allows us to trace the drainage system here. So now we're going to get into some fun parts where we get to look at some data, but first I need to get on a soapbox about interpreting data. So every time I'm going to ask you to look at data, I'm going to ask you to do it in 2 steps. First, describe the data and then interpret. Okay, this order is extremely, extremely important to develop your clarity of thought. Okay, here we're learning how to be better scientists. So what you should do is just tell me what the data look like. Just use your eyes as if you had no biology education at all. Just tell me what you see in the picture and then interpret. So I have a quick anecdote as to why this is very important. So I was hanging out with some of my psychiatry friends, psychiatrists, and they're talking about medical students and sort of the troubles they're having with learning. So learning is not the same thing as thinking. Thinking is an entirely different skill set than learning, So medical students are in fact fantastic learners. They absorb knowledge like sponges. They work very hard, but thinking is a whole different skill set, right? Anecdotally, a lot of them struggle with the actual thinking part. And, in psychiatry, this is especially important because, a psychiatric patient might not be in the right state of mind to give you accurate information. So you really have to think very critically about evaluating psychiatry patients. So we are not really taught clarity of thought in a deeply meaningful way in school. You get a little bit of it in your lab courses, but not in a really structured kind of way. So I illustrate this just to show you it is important to be able to do this in a real world context, because a patient will present with a reason for being in a hospital and a medical history, and whatever additional information you can investigate, right? And so a doctor must arrive at a conclusion, which is an interpretation of data, which is an extremely high level of complexity task. Right. But as as I was chatting with my psychiatry friends, students will often just vibe check a diagnosis without thinking things through. Like they'll be like, oh, this looks like bipolar, this looks like depression, but they won't actually think very carefully about whether or not this is true. Because, for surface level learning, you recognize a list of symptoms and then you just go, okay, maybe this is bipolar. But because of the nature of having a real patient in front of you, this often gets a lot more difficult when you have to do it yourself and when you have to conduct an interview, right? Because you get a tiny taste of the information when they're in the hospital, but it is up to you to critically think about where to go from there, right? So to combat this, clarity of thought must be established. So before you can, as a sumo wrestler, throw out, do a throw on a 400-pound man, you first need to do your leg exercises, right? I believe this is called shiko. This stomping motion that sumo wrestlers do to strengthen their lower body, right? You need to do the basic exercise in order to be able to do the fancy stuff, Before you can learn to draw anime, a lot of art students, when they're young, complain about like, oh, why does my teacher make me do X, Y, and Z? Before you can learn to draw anime, you need to learn anatomy, you need to learn posing, etc., etc. And before you can interpret data, you first need to be able to describe data, right? The foundation of thinking. Interpret data after you have been able to describe it. So now with that out of the way, did their engineered mice make a ZS green? So when they're walking through this paper, they engineered this mouse. Now we are trying to evaluate whether or not these mice successfully made their green fluorescent protein. And so let me get some definitions down before I ask you to be the scientist. What are we looking at today? We are looking at a Western blot. This is an experiment that gives you a semi-quantitative, don't worry about what that means, measure of protein amount. So proteins are run through a gel from top to bottom, transferred onto a paper, and then visualized with an antibody against the protein that is used. So basically what you need from this is where there is darkness, there is a protein. And the darker that protein is, the more protein there is, right? So some definitions: WT, wild type mouse injected with AAV synapsin Cre. So this is the wild type mouse. So this is not a special mouse. It doesn't have the green protein gene, but it was given the molecular scissors anyway. So this is a controlled experiment, right? These two mice are identical. These two groups up here are identical, besides the fact that one has the green gene and one doesn't. Sin ZSG, this is the ZSG mouse injected with the AAV. So they were given the scissors. KDA, this is kilodaltons. This is a measure of protein size. ZS green, that's our fluorescent protein. And nu N, that is a neuronal marker to normalize for total protein amount. You don't really have to worry about that for this particular figure, unless you're a true molecular biologist, Hello, I joined late. What's the end goal of making these mice create this protein? To be able to trace their drainage system. So those neurons are going to spit out the green protein, and that's going to help us trace the sewage system of the brain. Okay, so with that in mind, first, chat, describe the data. Don't make a conclusion yet. Okay, for this one, we're going to do it in two parts, right? Describe the data. So Neko, that is going to be an interpretation. If I ask you, did their engineered mouse make ZS green, and you say yes, that's an interpretation. But before that, describe the data. What do you actually see? What do you actually see? Tell me what you see. Pretend like you're 8 years old, you don't know any of these fancy biology words. Just tell me what you see. Give you guys a bit of time to do that. What do you see? So Vietran, wild type mouse, biomarker for ZS green, to no. Go a bit more simpler to that. Ari says huge black blot on ZSG mice and none on wild type mice. That's what I'm looking for. It's that level of detail. That level of detail. Just describe what you see in the figure. Wild type doesn't have protein, ZSG does. Treb, that's too far. We need to go simpler. Describe just the data. Describe the picture. Don't do any level of interpretation. Blobs or no blobs. That is the level that I need you to be at because you are skipping ahead of time. Okay. Awesome, awesome. Why does new n show no visible change though? So new n is just there to tell you how much total protein is in the sample. That's a what they call marker. I forget what the fancy term for it is, but it basically tells you, is semi-quantitative, right? So, in one lane you might load a little more protein by accident, and the other lane you might load a little less. New n is there so that you can do math to normalize the protein amount across all samples. So, yeah, okay. Housekeeping. Thank you so much, Micro. Yeah, housekeeping, Gene. Both mouses have neurons, but only one has ZS green. So that's an interpretation as well, right? I'm just asking for blobs right now. Just asking for blobs. It's for control? Yeah. You could think of it kind of like a control. But not quite. It's there for math reasons. Okay, so I'm going to model my expectations for you now that you've done the exercise. Some of you have already given the interpretation anyway, so I'll just do the whole thing. So the data, there are dark bands in the sample lanes from Syn ZSG, but not in wild type mice. So there are bands versus there are not bands, right? That's the level I'm asking you to do this at, right? Do not give me the conclusion. Give me just what your eyes see. And then interpretation is Syn ZSG mice are in fact making ZS green. No blob, blob, no blob, blob. Exactly, Paul. That's what I want. No blob, blob, no blob, blob. That's the description of the data. And so why do I ask you to do this? This is because if you vibe check the interpretation, you aren't being truthful to the data. Just like how a medical student might go, oh, this feels like bipolar disorder because I recognize the symptoms. That's not doing a rigorous job. You need to be able to fully understand the data before you can make the interpretation. Again, it's just like learning anatomy. It's just like doing your leg exercises. It's foundational. It might feel dumb, but trust me, if you learn how to think clearly, science becomes a lot easier. I do wonder if that line on the left, Syn ZSG, implies some kind of procedural error. It's just overexposed, maybe. It's just messy antibody, perhaps. Sometimes this thing. It's not a great Western blot, but it's also not horrendous either. Okay. So it looks like they did successfully engineer their mouse. At least this is one measure of them doing. We don't have another way of looking at it, but we do know that this mouse is making this protein. Whether or not it's making it in neurons, we don't actually know yet here. So let's go with a visual, right? So this is now a slice of the brain, and then they lit it up with fluorescent light to see whether or not they successfully made ZSG in their neurons. Very pretty image. So, PBS wild type. This is wild type mouse injected with salt water. And then the bottom one here is AAV HSyn Cree ZSG, right? This is the green protein making mouse injected with the molecular scissors to activate that gene. And then DAPI in gray, that's all the cell nuclei. So again, you can do this in the same sentence. You don't have to break it up anymore. But first, describe the data, then interpret it. You can do it all in one message. You don't have to break it up. But tell me what you see, and then tell me what you interpret. And the question here is, do we have neuron labeling? Do it all in one sentence now. Describe what you see with your eyes, and then give me your interpretation. I see the brain of the mice injected with PBS appear gray, while the brain of the mice injected with AAV Cree appear green. That's an excellent observation. That's exactly what I would agree with that as well. So, what would your interpretation be? Now, what is your interpretation? The bottom brain is greener, but is also more squished. Is that significant? No, just different brains. PBS no green, the other has green. Great. Yes, exactly. I think that is all accurate for your interpretations, your descriptions. But what about the interpretation? How would you do this if you are colorblind? Yeah, journals are not great with the color blindness thing. Is there color blindness that allows you to? So I'll say this for our colorblind folk here. I'm kind of ignoring the magenta here. So as long as you can see a difference between the top and bottom images, you're probably seeing the image correctly. Because I thought color blindness was more about contrast between similar colors. Because the only two colors in this picture are in fact gray and green. There's a bit of magenta, but that's not important for us today. Not everything is green. Different parts of the green brain are differently green. Yes, yes, yes. But what is the interpretation? Answer the question. Do we have neuron labeling? Are neurons labeled here? Are neurons labeled here? Are neurons labeled here? Are neurons labeled here? Oh, we have quite a disagreement. Okay, let's go into let's start a poll. Do we have neuron labeling? Yes, no, and then we'll go unsure if you're not sure. Okay. I'm seeing a lot of disagreement here. Do we have neuron labeling? Do we have neuron labeling? Do we have neuron labeling? Okay. So I'll stop when about half the people who are watching answer. Do we have labeling? Okay, that's just about half, and I will end the poll. So we have a bit of a mix. We have a bit of a mix. Half of you said yes, and then the rest are split between unsure and no, which is curious. So, what do I say? Our journal club streams a regular thing. I try to do them at least once a month. So, it's a trick question. You can't tell. So those of you who answered unsure, you are correct. So assuming makes an ass out of you and Ming, right? Why is this a trick question? Because, and this is why it's important to do this in 2 steps. Here's the data. The whole brain is lit up with green. So our interpretation should mean the whole brain is making ZSG. We have no way of knowing right now that these are neurons because there are other cell types in the brain. Do you guys see now why I ask you to do it in 2 steps? Does that make sense now? Does it make sense why I ask for the observation first and then the interpretation later, right? Observation first, interpretation later. The data show that the whole brain is lit up in this picture. So the only interpretation is that the brain is in fact making this protein, but we do not know if these are neurons, right? We do not know if these are neurons. We just don't know. But you also didn't specify the question that only neuron cell is labeled. So that's on purpose, right? Because the authors say, we use the synapsin scissors, so, only neurons should be labeled, which is true. Only neurons should be labeled, but this is a part of being a skeptical scientist, right? This is actual skepticism is, okay, you say your tools work the way that they should, but I'm not going to buy that until I see the proof for myself, right? That is the part of the critical thinking, right? This is why it takes so much longer to do journal club than it does for a video, because I'm sniffing through every single piece of data, and I'm not gonna, as my advisor would call it, drink the author's Kool-Aid. I'm not gonna just agree that we have neuron labeling. Okay, so let's move on then. So don't assume, right? Do the interpretation, do the description and then the interpretation. That's why we do this. Okay, so now we have a different picture. Now we're zoomed into a slice, okay? Zoomed into a slice. Now we have new N. This is our neuron marker. So this tells you the location of the neuron. And then we have Cre-AAV infected cells, right? So we'll go top first. All right, so new N, this is our neuron marker. ZSG, this is our fluorescent dye. And then you don't have to worry about GFAP or CD31. Just pay attention to these four. So Cree, this is an indication. These are the cells that have been infected with that molecular scissor. And again, this is ZSG. So now that we've zoomed in, let's do this exercise again. Describe the data and then make your conclusion. Describe the data and then make the conclusion, right? So what are we seeing here? First, tell me what the picture tells you. Basic language, you're 8 years old. Is the top half the uninfected mice? No, they're also infected. They're just labeled differently. There's fewer neurons in the bottom left. I would not really... So there's a good reason for that. So that AAV is not going to be able to infect every single neuron. So the Cree, again, just tells you which cells have been infected. Okay. So what do you see? What do you see? What do you see? And now you can look at this column, but ignore the red. Don't bother with the red. Don't worry about these. All right, so use this column to give you whether or not you see that neurons are in fact making ZSG. What is your interpretation and your observation and your interpretation? Observation and interpretation. Lots of green, but bottom green is dimmer. You don't have to worry about that. That's just regular differences in image collection. Yeah. So I'm starting to see some good observations in chat. Marlon says ZSG is all over the area. That is true. And, you know, I think that makes sense because ZSG is supposed to be spat out as well. So the background is supposed to be high. At least I think so. Okay. So what is my observation? What is my conclusion? So the data here are new N and ZSG have a lot of overlap. Right? You can see in this column here that there's a lot of overlapping green. You can kind of see it as white. So sorry if you're colorblind. It's very unfortunate. But this white overlap between green and magenta is indicative of the signals overlapping. So it looks like the same place that makes the neuron marker is the same place as the ZSG marker, which you can interpret as neurons are likely making ZSG. So this kind of evidence for a molecular biologist is about medium level proof. It's about medium level proof. It's like pretty good, but it's not definitive, right? This overlap could be a coincidence. It's probably not, but it could be, right? This is the nuance of science, right? This is medium level proof. Does that make sense, chat? Mullen says ZSG does seem to be concentrated in most where the new N is. Yeah, exactly. We're looking at the overlap, right? It doesn't look like every neuron is making ZSG, right? Like this magenta cell is completely devoid of green, so it's probably not making it. But that makes sense too, because like if you see Cree, as some of you have pointed out, this molecular scissor is not actually making it into every neuron, so that's probably actually fine, you know? Why are neurons circle? Aren't they supposed to be long? So it's just whatever marker. The new N, I think, stays closer to the cell body. It doesn't trace the entire axon. It's just the choice of marker that they used. Does the cream marker mark exclusively neurons? It marks the cells that have been infected. So it doesn't exclusively do neurons. But only the cells that are neurons are going to cut out the stop signal and then make the green protein. So it's a very nuanced answer there. I do wonder why it's in more than just the Cree cells. The ZSG, it's because ZSG is spat out. And it's actually an excellent question to transition to some future slides as well. Why are neurons making ZSG even when they're not infected? So I actually see pretty good overlap between the two, although it's not entirely obvious because this staining here is so... Sorry, I should be doing it here. Because this staining is so faint. Let's see. So this one... Oh, I think it's because the ZSG is leaking. Because I think everywhere you have a green dot, you also have at least a faint magenta dot. Like this faint magenta dot overlaps with this green dot, but it is quite faint. It is quite faint. That's a good observation, though. But here we have a little. Okay, so it might not be making the ZSG. It could be excreted, but that's an excellent observation to make. That maybe you do have some amount of funkiness happening, because this is a patch of green that doesn't really have. I mean, maybe it's this patch. It's hard to tell with the fluorescence, but it's good skepticism. Good observation. Okay, let's move on. So, as Marlon is pointing out, and others of you have pointed out, there seems to be a lot more ZS green than just in the neuron cells. And that could be because it's secreted. But again, microscopy is like medium level proof. We need something better, and that better is going to be a Western blot. So let's do this one again together. So if you just tuned in, a Western blot is a protein quantification method. So what happens is you run proteins through a gel, separates them by size, and then you transfer those to a paper. You hit them with an antibody against your target protein of interest. In this case, it's ZS green. And then the darker the signal is, the more protein you have. So in this left side, we have extracellular vesicles, EVs. So these are little fat packets that the cell spits out. And then soluble, this is water soluble. So this is just raw protein that the neurons, at least they think the neurons are spitting out. So again, first observation, then conclusion. Is ZS green secreted is the question that I am asking you. Is ZS green secreted? First, make your observation. Then make your conclusion. First observation, then conclusion. What are we looking at here, chat? Western blot. Classic Western blot. First observation, what do you see? Then give me your conclusion. What are we thinking about here, chat? Bit quiet. That's kind of interesting. Okay, we have some observations coming in now. No blots at all in the wild type mice. Yes, blots in the ZSG mice. Okay. Wild type barely black. Yeah, those are good observations. But what about your observations with regards to whether they are secreted? Because they're definitely true. Those are definitely correct observations to make. But couch your observation more in the language of EV and soluble. Observation: ZS green in the EV and soluble of wild type appear relatively faint, whereas the EV and soluble of ZS Sin ZSG mice appear to be intense but shorter band length soluble. Yeah. It's a good observation. What is WT wild type? So that's a normal mouse with no genetic engineering. Looks like something is suppressed for the ZSG to express. See, that's an interpretation. I want the observation first. Observation first. How often do we have journal club? About once a month. There's ZSG and EV and soluble fraction, so it could be secreted. I do the language first, right? It's very important that you describe it very simply. Even though I agree with you, train yourself to use the language of observation instead of interpretation, because there is ZSG and EV and soluble is the language of interpretation, not observation. Why does the fourth wild type have some black? It could be the antibody is a little funky. It happens normally. I wouldn't worry too much about it. There is some staining with the antibody in the wild type, but that's okay. It could be the antibody is a little messy. ZSG is secreted in both ways. Not really sure what these words mean. So EV is extracellular vesicle. It's like a little packet of fat that neurons spit out. And these protein samples are from those little fat packets. Soluble is like the water surrounding the neurons. We see more blobs in ZS green band and ZSG in both EV and soluble, therefore it is spitting it out. You see, that's kind of the way I want you guys to word your answer, right? First, the observation, you just talk about blobs, and then give me your interpretation. I can't see much of a difference in total protein in pictures. Yep, that should be fine. You know, this is, again, this bottom segment is usually just for math reasons, not too important for us, but it is nice to see. Okay, so how do I word this? So my data observation is ZS green stains heavily. So there's a lot of signal in this portion, in the EV and in the soluble, right? So notice how I'm just describing it. It stains heavily, right? I'm just saying it's dark here. And it's also dark here. That's where you should begin. That's the first thought that you should train yourself to have before you jump to conclusions. Then the conclusion I'm making is that ZS green is in fact secreted because there's a lot of this protein that shows up in the little fat packets that the neurons are spitting out. And there's also a lot of this protein in the water surrounding the neurons in the soluble fraction, right? So that is how I am trying to get you to think about interpretation, right? 2 steps, always 2 steps. Don't skip, right? Don't skip. Okay. So where does the ZS green go? So I'm pretty happy with the proof that ZS green is being spat out by something, right? So to sum, we can label the brain genetically instead with a brain injection. So we can do this, right? Whether or not it is physiological, as many of you have expressed skepticism over, that is a question that we have to think about and keep in mind. But we can do it. Whether or not it's accurate, we don't know yet, right? The label is made by neurons, is spat out by neurons. So if we're studying the brain's drainage system, we have to track where that spat out that protein is going, you know? So let's revisit this. So where is this protein going? So now we can talk about what this red is. And so sorry that if you are red-green colorblind, because this is probably really annoying, but I didn't make the paper, unfortunately. So GFAP, this is a protein only made by astrocytes, which are another type of supporting cell in the brain. CD31, this is made by vascular endothelial cells. And IBA1 is made by microglia. So the question that the authors want to ask is ZSG taken up by non-neuronal cells? So does it get absorbed by other cells here? Okay, I will address this because it's important. If schools can teach us how to think instead of diversity. Okay, soapbox. Diversity is good, actually, especially in schools, okay? It might feel fake and artificial to you, but this is very important that you understand this about me, is that I am a very strong proponent of diversity, especially in STEM labs, because research has shown that having a diverse set of people with a diverse set of backgrounds means you get a diverse set of opinions. And when you are doing rigorous science at the highest level, you need to be able to check all of your bases. If you have the same copy of the same researcher 8 times in a laboratory, you are going to get the same interpretation, the same results, and you are not going to be able to innovate as well. Research benefits a lot from international cooperation, from cooperation across different strata of socioeconomic circumstances. Diversity is unambiguously a good thing in science. So I will be having none of that language here. Okay. Carbon, this is your last warning. They shove it down our throats when we're like 10. Okay. Before I time you out, if you are going to be pushing this subject, think about why it upsets you so much and reflect upon that. Why does it upset you? And just stew in that for a bit, because this kind of thing makes me very upset. Okay. So, is ZSG taken up by non-neuronal cells? Again, give me your observation and then give me your interpretation. Yeah, let's get back to the data. Let's get back to the data. Back to the data. Is our soluble dye, the dye that's being spat out, is that being taken up by cells that are not the neuron? And tell me why you think that, you know? Tell me why you think that. Overlapping red and green signal create yellow signals, right? Yes, exactly. So wherever you see yellow, that is the overlap of green and red. Okay. There's a bit of overlap in GFAP, a little overlap in CD31, but in the IBA1, I think there is only little to no overlap. Yep, that's a great observation, right? Great observation there. So now tell me your interpretation. If you see the overlap, is it taken up by neuronal cells? Is it taken up by neuronal cells? Do we have uptake of this dye by cells that are not neurons? But yeah, I think Wei Chen, you have the exact correct observation that I think the authors are going for here. So with Wei Chen's observation, you can now go into an interpretation. So not so much yellow, so I would think that ZSG is not taken in. Yeah, you could say that. You could say that you're surprised by it not being taken up. You know, so this is great, because this is an interpretation of different thresholds of opinions here, which I think is really, really interesting. So what do I think? So it looks like there is uptake of ZSG in astrocytes, right? Because you see all these yellow dots here. There aren't a lot, and some of you have pointed out in chat, like it's not a lot, but it doesn't have to be a lot, because the question is it taken up, not how much is taken up. So here, because we have yellow here, we've got yellow here, a lot of yellow here, that's overlap of the astrocyte and the ZSG. It does look like it. Vascular endothelial cells, we'll go ahead and look at that too, and we do see, you know, yellow here, yellow here, yellow here. It doesn't seem as much. We've got a bunch of yellow here down as well. And then the IBA1 for the microglia, I don't really see any yellow in the IBA1. Those proteins really aggregate in the shape of arrows. My advisor would also agree that that's like a pet peeve of like putting arrows on pictures. You're covering data up, which is like not great. I personally don't like it, but it is occasionally helpful when it comes to subtleties here. So we have overlap, and again, microscopy, this is all medium proof strength, right? Medium strength proof, because again, some of this overlap can be coincidence, complete coincidence. So I wouldn't say yes, but I would say, yeah, looks like it. Looks pretty good, except the IBA one. I don't see the yellow at all in this one. Is it actually being taken up or merely diffused in? I don't really know if there's a difference technically. It looks to be inside of the astrocytes, looks like, from this data, right? Looks like. Is the picture colorblind friendly? No, unfortunately. Okay. So flow cytometry, this is a fancy technique that I'm not going to go too deep into, but the basics here is that flow cytometry involves separating cells one by one and then double labeling them. So this is a way to confirm. So fluorescence microscopy, that's like medium proof. This is like strong proof. Flow cytometry is like strong proof, okay? So we're backing up again. So some of you have mentioned, well, you can do experiments in different ways and try to confirm each other, make up for each other's weaknesses. That's what they're doing here, right? So they took the weak proof of the images, and now they're doing strong proof with flow cytometry. So they're going to label for these 4 different cell types. So microglia, astrocytes, and these are 2 endothelial cell types. And then they're going to double label them. So they're going to get hit with their natural label. So microglia is CD45, astrocytes is ACSA2+, BECs is CD31, BAMs is CD206. And then they're also going to get a second label. They're going to be labeled for the green protein, right, ZSG. And so what flow cytometry allows you to do is you filter each cell one by one. So each singular dot on this graph is the data for one cell. And flow cytometry will allow you to plot the amount of signal 1 versus signal 2. So the way that you interpret this is that these cells that are going straight up, right? These cells stain very heavily for the cell marker. So these here are like microglia for sure, right? These are astrocytes for sure. And then cells that stain very heavily on this axis, these are the ones that are that have taken up the green protein. They made the green protein. So what we're looking for here is a diagonal line. So if we see cells on the diagonal that have both a lot of astrocyte marker and microglia marker, in this quadrant, then we can be more confident to say that there are cells that took up the green dye. It's tough. This is tough. It's a bit of a tricky image to view, but what I'm telling you is that the more cells are in the top right corner, the more of that type of cell ate the green protein, took up the green protein. So what we can see is that there are, for every cell type, green in this upper right-hand corner, and a lot for the astrocytes. The astrocytes are like gobbling this green protein up. It's okay if you don't fully understand this. It's not very obvious, but you know, that's why I'm asking you to try it, right? If you can understand that this axis is like microglia marker, this axis is green protein. If you have both microglia marker and green protein, it means that this is most likely a cell that ate the green protein. Yeah. So much work went into building and validating the model in this paper. It's a Cell paper for a reason, right? Cell is a great journal. A great journal. So microglia does, but less. Yes, Viatran, that is the interpretation I would take away, right? Astrocytes, there's a huge cohort of lots of cells that ate up this green protein. And microglia, not so much. And that is reflective of this image, right? Astroglia, right? We see a lot of yellow here, a lot of yellow here, a lot of yellow here. But for the microglia, we don't see like anything, you know? What's gray? What's green? Gray is going to be the microglia marker here. It's going to be the astrocyte marker here. It's going to be the BEC marker here. It's going to be the CD206 marker here. And then the green is going to be the presence of green protein. So the gray means that these are cells that did not eat any of the green protein. Astrocytes are hungry, huh? Yeah, they're hungry for this green protein. They eat this green protein up for, you know, a lot, which is interesting to note. So here's just my explanation. Okay, yeah, the cells are on the diagonal, they're double labeled. So we've got a lot of astrocytes eating that green protein here. Okay, so non-neuronal cells are gobbling up the ZS green. So here we have the data. It's kind of hard to interpret, but you need to quality check this kind of stuff, right? You need to check to see if things are fishy or not here. And then this is like the summary of that data. So you can see that the microglia, a lot of them do have some green protein. Astroglia, fewer of them have green protein, but I think they eat a lot more of it as a percentage, right? Because this is percent parent. But here, you know, there's a bit of skepticism to me, right? Because I'm seeing that, oh, like this is suggesting that most microglia, the majority of microglia, have some green in them. But that doesn't square with this picture for me, right? I don't see any yellow on this picture. So I'm expressing some skepticism. This is why I take such a careful look at this data, right? It's fishy. And whether or not it's because I'm misunderstanding something or is it because the paper is fishy, that is what true skepticism is. That is how I'm trying to model skepticism for you. And so I have to think to myself, am I wrong or is the paper wrong? So I went back to the previous figure to look at it again. And then I came up with all sorts of different explanations, like maybe the process of flow cytometry opened up the microglia and then they ate a bunch of ZS green. So, you know, that maybe this data is bunk, you know, maybe the authors are wrong. But before I assume the author is wrong, I always try with 200% effort to give them the best possible chance of them being correct before I start flaming them, right? Before I start flaming them. And Frederick, you're saying, wait, maybe there isn't yellow because there isn't enough tissue slices to go over. And then Wei Chen says maybe there is, but it's really faint. That's all excellent. Excellent points. And then so what I did was I took the image and then I blew it up. And now I see the yellow. Now I see there's quite a bit of yellow. It's too faint and too low to see in the actual figure that they had shown, right? So if you blow it up, you can actually see that the majority of microglia do have some of that yellow, right? Yellow, 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 et cetera, et cetera. So wherever you see yellow, that is overlap of red and green. It might not look it, but microglia have this kind of like spindly shape. So even though this looks like it's in the green, this microglia is likely sticking a process over or under the ZSG neuron. So wherever you see yellow, that is where there's going to be a bit of that overlap. So it does look like microglia. You know, this looks consistent with the flow cytometry data. Looks consistent. See, this is why you actually quantify the overlap instead of just looking at it. Yeah, exactly. I'm not blaming the authors or anything, right? But they didn't have the quantitative data for this image. They just went for the flow cytometry, which I think is fine. I'm not really bothered by it. But it was really confusing. So yeah, it does look like microglia are taking up the green to some degree. It's just difficult to see. Okay, so let's recap here. Let's take a breath because there's a lot of information that came out all at once. So we can label a mouse's brain's neurons with a genetic method. That's the whole point of this paper, is a new way to do this. This is a superior method according to them because we don't have to directly inflame the brain by injecting something in it, nor push pressure through the vasculature. We ask the neurons to make a green fluorescent protein, which it then spits out, and then hopefully this can then enter the vasculature, and then we can trace the images of the brain. Okay, so how do these neurons hook up to vasculature? So right now we were just doing our confirmation or validation of the model. We haven't actually even talked about garbage collection yet, but now we finally get to talk about garbage collection. So Gizmo says they should have called it out in the paper. I don't know if they did. I didn't read the actual words because, you know, you'll hear this a lot from experienced scientists. After a while, you just look at the data and then you don't quite care about what the authors say about their data. I only look at the words the authors say if I have trouble reading the data. Like if I don't understand the experiment, then I'll read the words. But I actually just skim the words and I pay a lot more careful attention to the data. Because the data is where the story is. The author's words, they're like trying to convince you that they're right, and I don't like the coercion, right? This is the skepticism. You just look at the data raw. You read only what you need to get the context for what you can understand, and then you read beyond that if you want to have a conversation with the authors. So I'm very cautious in criticizing the paper, right? I think it's a pretty good paper. I don't express quite as much skepticism, although it is measured. I have a measured bit of skepticism here. Okay, so how are the researchers going to look at the vasculature here? So this is the system that they're using. So they are going to take their decapitated mouse. They're going to perfuse the brain with salt water. This is just to wash the brain of gunk that could mess up the imaging. Then they are going to label the vasculature with this reagent called WGA AF647. So this labels the vessels that drain the brain. And then they are going to fix the brain. So this is going to preserve the tissue with protein and paraformaldehyde. So labeling the vasculature is not the same as labeling the drainage, right? Because pipes can exist, but if no water passes through the pipes, then they're not really draining anything, right? So wash the brain, label the pipes, and then preserve the tissue and protein with paraformaldehyde. This is very standard. And then they do something called cubic clearing, which I've never heard of, but it's apparently a way to make the brain transparent. So it's a way to make the brain easier to image. And then they use something called light sheet fluorescence microscopy. Too long didn't read. It's a specific type of microscopy that allows you to illuminate thin sheets of the sample. So the goal is the same as confocal microscopy. If you're a molecular biologist, you for sure know what confocal microscopy is. Basically, this allows you to get 3D images, but instead of filtering in focus light like you would for a confocal microscope, light sheet, you shine a sheet of light through. So it's just kind of the reverse of confocal, but the goal is the same. Okay, so if you didn't understand a single word I said, that's fine. This is more for my biologists out there, right? You're using sheets to generate a 3D image, so now we can get a 3D image of the brain. And this is kind of, the thumbnail image, and this is beautiful. Look at this. Look at this, guys. Look at this image. Look at how sick this picture is. ZSG in green, obviously, WGA 647 being a label for the vasculature. ASMA, I think, is another label for a different type of vasculature, although, I'm not a brain anatomy guy, so I don't actually know the difference, but they're both like brain pipes that filter the brain. And they're so beautiful, right? This is amazing. Time to import that brain into Blender. I bet it would look sick in Blender. Is this different than wide field? It is. It is confocal. It's light sheet, so it's not confocal, but it also produces a 3D image. It's amazing. It's so cool. It is amazing, right? This is beautiful. It's 3D. It is 3D. I mean, they're not really gonna show us a 3D image of it, unfortunately, but if you know their raw data, they have every slice. So the way that the 3D image is made is that they are assembling a stack of 3D slices. That's what the light sheet microscopy allows you to do. That's amazing. Okay, so let's get into what what this can teach us, basically. So if we zoom in, we can actually see, again, green is the neuron label, and then we're actually seeing that at the interface of the vasculature, this double label blue and red, we're actually seeing little tiny blobs of that green protein, right? This is not neuron, right? Neurons are down here. We can see their cell bodies being labeled by the green protein. They're sending out their axons to the periphery of the brain to be drained. And we can see little packets of that green protein actually entering that vasculature. Again, microscopy is medium level proof. It's not great proof, but it is a start. So we can see that vasculature start to act as a drainage system here, which is just amazing. Just amazing. Okay. So the whole thing that these authors are trying to do is they're trying to make a name for themselves. They're trying to be the brain labeling guys. So they're going to try to compare their genetic labeling to the traditional injection labeling with OVA 555, so ovalbumin. So this is another non-reacting protein that has been conjugated with a fluorescent dye, 555 in this case, and they're going to do the thing that everybody has been doing up until this point. They're going to inject this dye into the brain, and they're going to compare ZSG to their injection. So ZSG is green and OVA is in magenta, and then the vasculature is in blue. So how does it compare, chat? Use your descriptive powers. Right? Tell me what you see. How does the green genetic labeling compare to the injected label in magenta? How does this compare? Tell me. This is a harder description, right? So far, the descriptions have been pretty easy. The data have been pretty easy to describe. But do you see why I train you to describe the data? It's because this is a lot harder to describe than the earlier stuff. So do your best, because it'll help you develop a language, a vocabulary to describe data. Aren't those angled differently? This is the same mouse. This is a double labeled mouse, and with two different perspectives, right? You can see this is a top down from the mouse up here. This is a side view. And this is the slice that they're taking. What would you say? How would you compare these two? RSY says some green alone and some magenta alone. Yeah. There's some green where there isn't magenta. There's some magenta where there isn't green. Now, tell me about what your interpretation would be. What is your interpretation? So the patterns are different, right? That's your observation. The pattern of staining is different. Magenta and green do not always overlap everywhere here. What does that mean? What is your interpretation there? What is the interpretation there? Maybe the genetic labeling makes the protein act natively while injection only goes up to a certain point. Possible. Possible. Magenta stays more towards the back of the head, potentially not fully diffusing into the brain. Also possible. It's unclear to me which regions are more important to highlight. That's an excellent observation to make, Marlon. But at this stage, we're value neutral here. We're value neutral here. We're just being descriptive. The injection messes with the drainage system. That's the interpretation that the authors want you to have. The authors would love it if you believed that is the reason, right? But it's complicated. The injection stays more local. Yeah, that's something also that the authors want you to believe. I'm not saying you're wrong, by the way, Ari and Syra. You're not wrong. It's just those are the interpretations that the authors will lead you to believe that that is true. Okay. So the labeling is quite extensive genetically, is what I would say, right? There's a lot of green here, a lot more green than magenta, it looks like. But the injection the pattern of expression is quite different. I'm not saying better or worse, right? I'm not making a value judgment, as Marlon is careful to point out, right? Like, I don't know which is better, which is more accurate, and we can't know which is better, right? Because that's the thing we're trying to figure out, is which is better. And I don't think we're going to get a very satisfying answer besides this interesting finding that it is different. Injection is different, and it does like injection accumulates much more in these regions. This DCLN, this is a lymph node, and it looks like when you inject this, it accumulates in the lymph node a lot more than the green would. So at this stage, we don't have enough information to know which is better, just that they are different. Okay, and then they take a look at the different brain regions across different border compartments, and then they compare the level of staining between the ZS green and the injected. So injected in magenta, green and green, obviously. And you can tell that we're getting a lot of differences. The pattern is really different. You can see that, like, even though they're not making direct statistical comparisons, you can tell that the ratios are different, right? Like, there's so much in the brain in the green, and then there's not a lot in the lymph nodes. And in the magenta, there's a lot in the lymph nodes, but not a lot in the brain. So again, this is value neutral. We don't know which is more accurate to the real brain, but we can tell that they are different. How did the genetic method work? They just delivered the gene into the brain. The mouse is a genetic engineered mouse that is that has the green protein gene blocked by a floxed gene with a stop sequence in between, and then you inject an AAV to cut out the stop sequence just in neurons. That's a high level explanation there. Yeah. So it is interesting. It is interesting. It is different. It is different. And then they're going to take another look at some deeper regions into a brain, so going more in the middle of the brain to take a look at these differences. And as your observations have made known before, there are regions of green that don't have magenta and regions of magenta that don't have green. So here we have regions that are genetically labeled, but not injected labeled, so green only. Don't know why they circled this one, because this clearly has both, so I'm not entirely sure what that's about. And then here you have the opposite finding, where you have a lack of genetic labels and you have a presence of injected label. So they want you to believe that these are false negatives and these are false positives. So it is interesting, right? It is interesting. Okay, so genetic labeling and injected dye have different distributions. So what does this mean for drainage? And I think I need to address this before we move on, right? So I don't think epistemologically there is a way to prove that one is better than the other beyond the method argument. At least I'm not clever enough to think of a way where it would be more or less accurate. But we will see one layer of proof in a little bit. Okay, so I'm delaying it because, again, this is me being biggest brain possible, being as skeptical as possible. We don't know which is better. They will make an attempt to argue that their way is better in just a little bit. Okay, so what they're showing here, this is the eye cup. We're zoomed into the brain. This is where the eye socket would go. And there appears to be, only with the genetic labeling, a lot of drainage into the region beyond the eye cup in the zygomatic, which is the region that drains below the eye, which is really interesting. You also see regions that are missed in the nasal cavity. So here you see green. This is actually really good, though. Like this makes me believe their data more, because you see the subtle presence of drainage into the actual vasculature itself, and it looks like the injected dye can't quite make it all the way out to that periphery. Same here. A lot of green where there is no magenta didn't get into it quite as well. Okay. And then same with the dorsal dura and skull, just showing that we're getting a lot more fine drainage of that green protein than we would magenta. But okay, here's where we're going to address this elephant in the room, right? So I think people have already, I'm not going to bore you with this explanation, but people have already been chatting in chat. You guys are already one step ahead of me that, just because this genetic method seems to work more, as in you can stain more, that doesn't necessarily make it better or more accurate to the true human brain, to the true mouse brain. So how do we know that the genetic label ZSG is physiological? Like how do we know that it actually reflects the real brain structure? No matter what, ZSG is just one protein with a specific size, a specific shape, a specific polarity, a specific osmotic pressure, right? The gene for the green protein is not this ephemeral thing that doesn't interact with anything biologically. It does. It interacts with stuff. And so the question that I have is that, are we studying brain drainage or are we just studying ZSG trafficking? This is a big point of skepticism for me. Do you have any ideas on how to address this issue? I didn't. Full transparency. I had no idea how to solve this problem. It's a really hard problem to solve. And it's a problem that has been haunting us since the beginning of this paper. Are we just looking at ZSG trafficking? We genetically made this protein and we're watching it flow all over the brain. Are we watching real brain drainage? Are we getting a complete map of the sewer? Or are we just watching where this protein goes? Query, what if they did this as a time series to see what parts are at the start and what parts are at the end of the brain's drainage system? I don't remember if they did. It's an excellent question. Whether or not they did a time series, it would be difficult to do in a live animal because you need the animal to be alive. I'm guessing they didn't because live imaging in animals like this to this level of resolution is pretty hard. I think it's possible, but I'm not sure every lab is equipped to do so. Much easier to image a brain that has been fixed and treated with formaldehyde than it is to do in a live animal. Are there any other dyes that work like ZSG but with a different structure? Do it again but with a different fluorescent protein. These two get the same strategy. Why not just choose something different? And it's okay. Those are okay, right? Those are okay answers. But then you just run into the same question of, well, are we just tracing one other protein? We could use proteins of different polarities and molecular sizes. Again, it's the same problem. It's the same problem of like, if you just pick another label, you just run into the same issue. Are we just studying the trafficking of a different label? It's interesting, right? How are they going to get around this? How are they going to get around this? I could not come up with an answer. This is where I really appreciate the genius of this technique. So what if instead we used a couple of different proteins? What if we used every protein? Think about that, chat. What if there was a method, instead of one protein being labeled, what if we could label all of the proteins? Think about that for a second. What if we could label every protein and watch every protein move within the neuron? Wouldn't that be something interesting? Wouldn't that be interesting? And that is a lot better than trying one or two proteins at a time, right? It's a lot better. You ohm disrupt, yeah. I'm a doctor and I love your content. Thank you. I frequently use TC99M HM POW for brain death perfusion scans. Oh, interesting. Passes the BBB and remains trapped in functioning neurons. I actually don't know that technique. I'm not sure if they use that here. How on earth would you label all of the proteins and how they move? Ah, right. And I'm letting you sit in this mystery a little bit so that when I reveal their technique, it's going to be a lot more amazing to you. So we're just going to sit here just a little bit. We're going to try to label all the proteins. How are we going to do that? It sounds extremely expensive, cheaper than you might think. Okay, so how are we going to pull off this magic trick, huh? This is why I love science so much. This is so cool. And the answer is actually so simple. And those are my favorite moments, right? This is a very simple solution to a very difficult problem. New proteome labeling. Just as Brady said, Brady is cheating because Brady has a PhD in biology. So yes, Brady has the correct answer. New proteome labeling. New proteome labeling. So we are going to inject intraperitoneally, so right up the hoo-ha of the mouse, with a dye, AZF674. This is a blood-brain barrier permeable non-canonical amino acid with a fluorescent label. What does this mean? AZF is a tryptophan substitute. So proteins are made out of amino acids. One of those amino acids is tryptophan. AZF looks a lot like tryptophan, but this AZF is modified with a fluorescent label on it. Or I think maybe it's fluorescent by itself. I'm actually not sure. I don't know too much about AZF. But yeah, now the O's are coming out, right? Because what does that allow us to do? Yeah, a lot of oohs and ahs, and then the eureka moment, right? What does this allow us to do? If proteins are made out of amino acids and you incorporate a non-canonical amino acid that is also fluorescently labeled, all new proteins in the brain are going to be fluorescently dyed. New proteome labeling. So cool. Right? Do we get that? Do we understand? I need to pause here. Do we understand this idea? Do we understand new proteome labeling? Because this is a big eureka moment. This is going to allow us, in theory, to circumvent the problem of like, are we just looking at green protein drainage and not the true sewage? But if we can label every protein, then that solves the problem. Doesn't that mean you miss proteins that don't use tryptophan? You do. If they don't use tryptophan, you won't see them. But the idea here is that you capture enough of the proteins to get an idea for how most proteins will drain. Therefore, the observed pattern is unlikely to be caused solely by ZS green because they compared with other synthesized neuronal proteins. Yes, newly synthesized neuronal proteins, fresh ones. Would the new amino acids interfere with protein function? They might, but the idea is that you pulse it and you don't let it go on for too long so that most of the proteins in the body are still fully functional. Again, what you're getting at, Thor, or you're getting at, Hattie, is no experiment is perfect. Because in order to study something, you have to modify it. You have to label it. You have to measure it somehow. But you want to minimize the impact. And you want to compare between two different methods, right? This has weaknesses as well. As people have been saying, you know, those proteins that incorporate this unnatural amino acid are going to be a little messed up. Or maybe even very messed up. But the idea here is that if this AZF labeling shows the same result as the green protein labeling, then we have a much stronger level of evidence, right? I'm trying to impress upon you that science is all about levels of proof, confidence, right? It's never true or false, it's confidence. And if these two things give us the same result, I'm going to be a lot more confident, right? How can we differentiate between different proteins if that's the case? We don't need to. We're just looking at drainage, general drainage. So, this is fun. This is so much fun. Okay. So, qualitatively, this looks similar, right? Unfortunately, you know, I was saying, talking about big game, if we compare these, then they're great. But unfortunately, we can't do this in the same animal. Since the AZF also uses this AAV-Cree technology, it would be pretty complicated to get all of this working in the same mouse. So instead, we have to compare it between two different images. I'm a bit sad because the best level of proof would be to have this in the same mouse, but instead we're just going to have to go with what we've got here. So I'm actually going to take a quick restroom break, but I'm going to let you stew on this image. And make the observation, right? Like really look into this image here. I'll actually make this a bit bigger so that you can see the images a bit bigger. And I will be right back so that I can do the rest of this presentation with no interruption. So BRB. All right. Okay. Yeah. So, let's go ahead and shrink this again. Very beautiful image. Okay, so in magenta, we have the method by which you are labeling all the new proteins by incorporating this unnatural amino acid. So are we looking at the same mouse in two images? No, this is a different mouse. Different mouse. Yeah. Ba, ba, ba. I would want to compare against a variety of proteins, not just one subset. It's not. It's like a lot of proteins is the thing. Okay, so I want to take this opportunity to give you guys something that my advisor taught me when I was in graduate school. It's very easy to be critical of a paper and constantly say that it's not perfect. So for chatters who are skeptical of the paper, and you're okay to be skeptical of the paper, try to follow up the thought with a way to improve the experiment, right? And by doing that, you can force yourself into their shoes and can kind of see why they did not choose certain aspects. So like if we're talking about whether or not this is improvement, I think this is a huge improvement, because in green we only have one protein, and here we have hundreds, thousands. Any protein that has a tryptophan, you get to see the vasculature here. Yeah. Okay. You can overlay the images with 50% opacity to see the difference. You could, however, it's not, that's actually worse. That's implying that they can be compared when you can't actually. Like, it would be irresponsible to directly compare these two. We kind of have to cheat to compare these, right? Because if you overlap them, that implies that it comes from the same mouse. So you actually can't do that in a way that is kosher, scientifically speaking. But we're doing it here just to get a feel for how close this is to the other technique. Okay. So how fast do these drainage systems work? So if we're making the assumption that these are working normally, and I do like this experiment of like doing the tryptophan substitutions, you pulse that in and then you label everything all at once. I think that is a pretty good way to do this. And now we get back to the You Be the Scientist moments. We got another blot. This one's a different blot. This is SDS page gel electrophoresis. So this is all of the proteins. So we're no longer using antibodies. This is every protein in this section of the brain. You isolate the different components, you grind them up, you isolate the proteins, and then you put in a blue dye, and then you take a picture. So what we're seeing is total protein in everything. And so the question I have for you guys is, how fast does this drainage actually go? So here's how the experiment works. You inject that AZF label. It's a finite amount, right? You don't constantly feed in the AZF. You give them a little pulse that labels some proteins in a window of time in their brain. And then after 7 days, you begin collecting data. So T0, this is when labeling is complete. And then T2 to T5, you have a washout. So time 2 and time 5. So that's what these are. Time 0 is when you start collecting that data after labeling is complete. And then T2 and T5, you withdraw the label, and then you can make the interpretations from there. And again, when you are looking at this data, make sure you first give me your observation. So describe the data first, and then give me your interpretation. Remember, a few days, like a week or two. I actually don't remember how long the washout is. That's on me. I don't remember how long. But we'll broadly characterize them as fast, medium, and slow. So, and I know that's not really scientific, but just to give you an idea, right? Just as an exercise, fast, medium, and slow. So, what's it look like? How fast are we draining here? First, give me the observation, then give me the interpretation. DSK is somehow increasing protein content. Interesting, isn't it? something weird is happening in the Dura. I observed that the bands in T0 is heavy, the bands in T2 is faint, the bands in T5 is more intense. It is interesting, and I'll address that. So every band here is a different protein, right? And so what you might be seeing is different proteins getting filtered out at different times. So even though the band is darker there, that might be because there's a different protein in that same molecular weight spot that happens to take longer to filter out for whatever reason. Yeah. DSK stuff seems to appear over time. LSK seems to get slightly lighter over time. Brain gets much lighter, stays roughly the same. Dura decreases, and S quickly disappears. Yeah. Gosh, I really miss looking at protein gels. They're so fun to discuss and theorize about. I know, they're so underrated. I love microscopy, but there's something epistemologically very satisfying about a gel because you get so much information at a strong level of proof, right? Because microscopy is nice and powerful, but it's not that powerful. Western blots and SDS page gels, these are really powerful. You get very high confidence when you do stuff like this. So, all right, so let's go through this together. So DSK, dorsal skull, how fast did this drain? It's slow. As Brady said, it looks weirdly like T5 even has more protein. Within these gels, because these each come from a different mouse, it could just be mouse-mouse variation, but these are about the same. This does look a bit more, but because it's mouse-mouse, there could be some variation. I would say that T5 is not lighter than T0, right? That would be my interpretation here. Did you see that Thermo Fisher just got caught out faking a bunch of the protein gels? I know. So sad. Yeah, it's just so sad. Okay, so LSK, lateral skull. As you guys have said, this is also slow, right? This is very similar to DSK. And then the brain, now we're getting somewhere, right? Now we're getting lighter in the brain, right? So the brain's draining, I'm going to call it as a medium speed, which I'm going to also say for the Dura as well, right? There's some drainage. And then we get to the nasal cavity and the cribriform plate, which is also where the brain-eating amoeba go, by the way. The CRP, I couldn't include that in the video because it just felt like auxiliary information, but the CRP is the region between the nose and the brain where the brain-eating amoeba like to go up. And these, I'm going to call them fast, right? Because as you guys have correctly observed using your observation skills, that these bands here are much lighter than this band, right? In T2 and T5, in the cribriform plate, this drains real fast, right? This T5 lane almost looks empty. So with this dye, we can actually learn how fast these different systems drain, which is a nice baseline to kind of figure out. Fast drainage in the nose is fitting. Yeah, I agree. It makes sense. It's consistent with what we know about the brain, the nose. So this is our sanity check. So this is the quantified data. I'm not just going to let you look at pictures. Let's actually look at the numbers. And here we have the DSK. There's no significant difference. It doesn't look like it drains very fast at all. And as Brady had pointed out, it looks like T5 even looks a bit larger, although the difference is not statistically significant across the replicates. LSK looks like it does drain, but it's not statistically different. And then the brain, you start getting some drainage, although this one at T5 doesn't look like a lot. Still relatively slow. But then if we look all the way to the cribriform plate, this is lightning fast drainage. That is a lot faster. And the nasal cavity as well. So nice. With this label, we can actually start to learn some basic information about the brain's drainage system, some basic facts. Okay, here we have a video. Annoyingly, there's no caption for this supplemental video. It's probably buried in the methods, but I'm already like wasting a lot of time, right? If you think about it, I do Journal Club for fun and to give my members the thing that they're owed, right? Membership, you get, if you are macrophage or higher, you get to pick what paper I do. But I'm gonna be like straight up with you guys. I don't do Journal Club because it makes me money, because I actually don't make money on these. So I'm just be real honest. I could have gone deeper here, but I was already behind and I didn't want to get even more behind. So I did not go to the methods to read what this was, but it wasn't in the caption and that annoys me. So Yeah, I'm just really annoyed because this video is cool, but I'm going to be guessing at what we're seeing here. So I believe blue is AZF, that's like general protein, and then the magenta I believe is some bone marrow marker. I think this is some bone marrow marker. So this is showing the drainage. And so what we're seeing, I believe, are cross sections. So when this animates, don't think of it as time. You know, we're not moving forward in time. We're moving through the bone. Okay? We're moving through the bone. And that explains why this has like a spongy texture, because the bone marrow is kind of spongy. So just imagine, you know, we're starting at one edge of the bone and we're moving through the bone. We're not moving in time. This is a fixed time section. Let's watch this video. Yeah, we're moving through that bone. And what do we see? I don't know why they do these zoom ins. Like, I don't know. They don't have any captions on this, so there's no explanations, but I'm pretty sure these are cross sections moving through bone. I think the video is so cool, but I'm so annoyed that there's no caption here. There's a point where AZF disappears. I think they just like remove the layer that shows the AZF. I don't actually know if that's meaningful. Again, it would be better if they had it in the caption. But what I'm taking away from this, can we replay this? What's going on? What I'm taking away from this is that there are streams, right? We're seeing streams of this blue protein run through the bone marrow in these lines as we're moving through. So you know, the bone marrow is also where there's blood, so it looks like this protein might be filtering into the blood, which is neat. Very, very cool. Very, very cool. Okay, here's a summary so far. I like to do these every once in a while just to pick people up in case they just tuned in. So we're doing genetic labeling instead of injection because hypothetically speaking, it should be better because you don't have to push pressure into the drainage system. You could trace drainage with these systems. We've seen it before. You can even see how fast different regions of the brain drain. So what else can you do? Well, you can see what cells are taking up the drainage, right? We saw a little bit of this before with the flow cytometry, but now we can really do this for a lot more cells, right? We got a little taste of this before. Now we're looking at a lot of different cells, and what we're seeing are clusters here. So, you know, to the right, if you see cells drag to the right, that means that they are taking up more of the ZS green, and then the gray up here is less. So basically, the longer this each like these are always so hard to explain. The more these blobs look pulled to the right, the more those cells are taking up the green protein. So this allows us to actually check who is responsible for clearing out these cells in the drainage, right? So before we were doing brain generally, now we're looking at dorsal dura, we're looking at the skull, and we're looking at the nasal passage. Hey, Fi's in one of these pics, exactly. I mean, all of them actually. The neutrophils right here, neutrophils here, neutrophils down here. I'm all over the place. So the more we're stretched to the right, the more we are eating up the green protein within these different regions. And I'm going to ask you, what is the most striking thing? Maybe what is the most surprising thing about this to you? I know that there's an observation here that I'm really, really surprised by. And what might that be? This is a hard level question, by the way. This one I only expect people with biology background to get. For most of you, this probably just looks like blobby nonsense, but really look at what the labels are and then compare them between dorsal dura, skull, and nasal. What is weird about this? What is weird about this? Question, if this fluorescent thing covers all proteins instead of just the green... Oh, no, we're sorry. We're back to the single protein. We're back with the single protein. Why were they faffing about with the green? I think the green gets you better resolution. The green allows you to trace a lot better. I think when you do the whole protein labeling, you don't get as much definition. And here we're getting a lot more definition. Honestly, I can't read. Okay, let me make this a bit bigger then. My OBS. Just temporarily going to hide chat. So that should be more visible. What is striking about this? Comparing between dorsal dura, skull, and nasal. Yeah, Microblog says it seems to be pretty different between the different locations. And I've noticed you probably do have biology background, right? So that's what I was saying. I think only my biology bros will be able to tell, but everybody's welcome to give it a try. Is green better because humans see green best? Oh, that's a question I can't really answer very well. So this is a microscopy thing. Okay, I think green... It's not because humans can see green better that they chose green, because we're not actually seeing the green. It's the microscope that sees the green. I think green is likely the choice because the microscope can see the green the best. I think that's the best way I can put that. Why do the same cells in different tissues have different uptake? Yeah. Yeah. This is a UMAP, right? This is a UMAP. Exactly. That is what we should be concerned with here. And again, don't worry too much if you can't understand this. It's not really worth going in depth as to what a UMAP thing is for this particular figure. But what I want to show you is that, you know, neutrophils are all the way up here. They're eating a lot of this green when they're in the dorsal dura. When they're in the dorsal dura. Or rather, they're eating a fair amount, I should say. I shouldn't go too overboard, right? But if we move over here in the skull, it looks like there are a lot more neutrophils in the skull, right? But they're not eating as much, relatively speaking. And then when you go down here, you see that the neutrophils, that there's a large variety, right? Some neutrophils are not eating a lot of the green at all, and then some neutrophils are eating a lot. And that's very different behavior than in the dorsal dura. So the striking thing here is that depending on which drainage system you're in, whether you're in the dorsal dura or directly in the skull or the nasal passage, different cells have different patterns of green protein uptake. Yeah. So even though the cells remain the same, the patterns are very different, right? Macrophages up here, you know, they're feasting. A lot of them are feasting on this green up here, but then they're like less hungry down here. Is the idea. Or rather, it's different because I don't quite remember what this ratio is. It might be the reverse, but what I want to impress upon you is that it's different between each of these. That the patterns, the blobby patterns, the Rorschach test are not the same between these three locations in the brain. Oh, we can't see your mouse. Oh, sorry. Yeah, there we go. Now it's visible, right? Each of these blobs, macrophage blob looks like this. It looks different in the skull, and then it looks different in the nasal passage as well. And that's striking because ostensibly they're the same cell type, right? Neutrophils up here, neutrophils are now up here, neutrophils are now all the way down here. They're different. They are different, and that is weird because it's the same cells, but depending on the context, they act differently. That's what I want you to get out of this figure. So now the mouse should be visible now. Okay. What else can we learn? Different parts of the brain have different immune profiles in response to protein flux. So if we're going to be leading up to this grand discussion about clearing the brain of garbage, we need to talk about the immune system. And so what we're also seeing here is that the immune profiles in response to protein flux are also going to be very, very different in these different regions. Dorsal dura, you see that there's a lot of endothelial genes that are differentially expressed, meaning there's a lot of up and downs in their different genes. But guys, look at the skull. Look at the skull. Tell me what's striking about the skull. Is there anything striking about what the skull is doing? What is the skull doing? What is the shocking difference in the skull? B cells. So, what does that mean? Hyper, yeah. Look at how crazy that number is. Almost 4,000 genes in the B cell that are being differentially regulated. They're either going up or they're going down. what is protein flux? Protein flux is just like the movement of protein in and out of the drainage systems. There are a lot of B cells, damn. It's not about the amount of B cells. It's that the B cells in the skull are differentially expressing almost 4,000 different genes. So the B cells are doing something. They're upregulating thousands of genes. They're downregulating 1,000 genes. The B cells are really different in the skull than they are in any other location in the body. That's what you should get out of this particular data. What does the up in ZSG plus and down in ZSG plus mean? It means that, okay, so say, for example, you have a normal mouse, and then you profile their B cell. You say, okay, B cell, you are making this gene at this level, this gene at this level, this gene at this level. And then you do it for all the B cell's genes, right? You get a full picture of what the B cell is making in a normal mouse. When you force them to make this extra protein garbage, that is our label, the B cell will make more of 3,000 different genes and make less of 1,000 different genes. So in response to this fluorescent label, now we're calling it garbage here. The B cells are really, really different. They're doing something really, really different here. Not only the B cells, but the GMPs, pre-B cells, Tregs, neutrophils, NKs, and eosinophils. Correct. There's a lot of activity going on when you force the brain to make this random garbage here. They're very active in the brain, unlike the dorsal dura nasal. I would caution about activity. It's more like they're doing something different, right? Activity is a different thing because they could still be very active. It's just that in this case, we're looking at a change in behavior because of these differentially expressed genes. They're doing something different than they normally would. And Viatran says immune monitoring. Yeah, maybe that is a hypothesis. So what is the B cell doing, right? 3,000 of its genes are going up, 1,000 of its genes are going down. The authors are really curious to know what the heck the B cells are doing. Making genes, you mean like what genes are used to produce proteins? Yes, correct. I simplified it for the sake of simplicity, but yeah. So B cells are the antibody producing cells, right? So what the heck are the B cells doing? So they took a look at all of the genes that the B cells are making more of and making less of, and then they did some fancy database, big data stuff to it. And what they found was an upregulation of pathways meant to increase tolerance of antigens. Tolerance of antigens. So you can see down here that a lot of genes that are associated with tolerogenic pathways are increased by a lot. And then the inflammatory pathways are decreased by a lot. So the B cell, it sees this green dye protein, and it's like, well, I gotta do something about that. And instead of attacking it, what the B cell is seemingly doing is teaching the body to tolerate it, right? Seemingly, and I use this very, very carefully, right? So in other words, B cells are monitoring the trash and ensuring the protein doesn't freak out about the protein waste. And it's important to know that gene expression data is only a hint and not hard proof of itself, right? I would never claim that the B cell is making it tolerable, because this is, if we're talking about the spectrum of proof, this is weak proof, right? It's proof, but it's weak proof. So it's important to note that ZSG is not an endogenous protein. Your body doesn't naturally make it. So this might still be a weird effect of the genetic labeling. But importantly, for this particular experiment, I don't see a non-AAV injected control, which screws with the interpretation of the data a bit that I'm a little too tired to explain why. I apparently knew why when I made the slide, though. Their fold expression color scale doesn't have white as 0. Oh, does it? That's so funny. That's so sad. That's pretty terrible. Yeah, Brady is right. This should be moved here. That might be a mistake. That's pretty crazy. What do the B cells and other bones do? Make antibodies. So it looks like the B cells in the brain are like, hold on, let's not make antibodies actually. Let's actually learn what this protein is and make sure that we don't freak out about it. Otherwise, we're going to cause an autoimmune reaction in the brain, and that would be very bad. Supposedly. Again, this level of evidence is very weak. It's not strong proof, but it is proof. Okay, so that's interesting. This bit is just going to be random things they learn about the brain. I don't think there's a coherent story here so far. Okay, the next question they want to ask is, how does the brain actually drain? Right? Does the brain enter, so you make all this green protein. They want to know like some basic biology of the brain here. So that's why this all seems kind of discombobulated. They want to know some basic facts about the brain. So they want to know whether or not like you see non-specific clearance, whether or not you get like waste that like enters and then goes all over the place and then exits, or whether or not the region itself is what drains, right? So the question they want to ask is, does waste in the brain drain locally or globally? Locally or globally? So that's something that you can do with this tool too that I think is pretty cool. We encounter a lot of novel molecules in everyday life. Does this have to do with why we don't have an immune reaction every time we encounter a new chemical? Yeah. So you have a lot of immune cells that teach your body to tolerate a lot of things, and when some of those things don't work as well, you develop allergies or immune reactions, right? That's a whole complicated topic that I'm not ready to dive deep into. It'll probably be its own like 30-minute video. So we don't actually have the answer to this. They're going to try to figure it out. Is it global? Like, does it circulate? Does it go onto the highway and travel all over the place and then get drained? Or does it drain locally and then exits the nearest exit? So they have some fancier AAV constructs that make ZSG only in certain regions. So now we are beyond making it only in neurons. Now we're only making it in neurons in certain regions. And this is with a much fancier promoter than just a broad neuron promoter. Now we have a forebrain promoter, so only neurons in the forebrain get labeled. We have an anterior brain, so this is the front of the head, and then the striatal brain label, right? So in order to test whether or not drainage is local, we have to first make sure the gunk only shows up locally. So very fancy technique. This paper is absolutely massive. I know it's a cell paper. This is what careers are built on, right? You get a cell paper and it's often said that your career is set. I do not have a cell paper. So it looks like ZSG drains in different locations. So again, we're comparing dorsal dura, BSK and dura, and then the nasal cavity. And we look, we're just going to go over this very quickly, but it looks like the distributions are quite different. It looks like in the dorsal dura that the forebrain gunk appears a lot more, and it's a lot less in the other regions. So it does look like in different regions, we have a different pattern of drainage, and that's all I want you to get out of this figure. So things we learned so far, we are reaching the end of this paper soon, I promise. Genetic labeling might be better than injection because you can see more things that are more likely physiological. More likely, we don't know that for a fact, but again, we have different varying levels of confidence. You can trace drainage systems. We can trace drainage with these systems. You can even see how fast different regions of the brain drain. And you can even see that immune cells are doing stuff in response to the ZSG label, and that ZSG drains locally as opposed to one big circuit. A lot of massive things that we're learning here. Basic stuff about the brain. Basic stuff about the brain. Okay. What else can you learn? Well, how does disease affect brain drainage? Again, when you are a molecular biologist, a lot of the money that you get to do the very expensive research that you do in the lab, $1 million to a lab is nothing. It really is nothing. Like $1 million in a lab will set you up for a very short amount of time. There's a massive amount of money. And as a molecular biologist, you need to think about what impacts your research has on humanity. And a lot of the times, if you study molecular biology, you gotta link it to a disease somehow. And this makes sense. I'm not saying this out of cynicism. I'm just reminding people that research is done for a purpose, and in molecular biology, that is most often to cure diseases and stuff, or to research different diseases. So how does disease affect brain drainage? They're going to look at two different models. One, LPS, lipopolysaccharide. So this is a sugar that that bacteria make that is highly inflammatory. This is how the immune system recognizes a lot of different bacteria. They sense this important membrane sugar and then they go crazy. LPS is highly, highly inflammatory. It is an SOS signal to indicate that a bacteria infection is underway. I've talked about LPS multiple times on this channel, in the videos of this channel. And this is most often a stand-in for general inflammation, right? People use LPS as a tool just to ramp up inflammation to see what inflammation does regularly, normally. And we're also going to look at a amyloid beta mouse. This is a mouse model for Alzheimer's disease. Me when I spend $12,000 on publishing a single open access Nature paper instead of valuable reagents for actual research. You know, God bless you. God bless you for doing open access. God bless you, Brady. Yeah. So, the whole paper is structured around analyzing or researching or talking about Alzheimer's disease. So that's why we're looking at amyloid beta. And again, I mentioned a bunch of caveats. Okay, like it's 2026. A lot of us don't buy that amyloid beta is the most important thing in Alzheimer's anymore, or the singular most important thing, right? I think the more nuanced position in 2026 is that it matters. We just don't know exactly how much it matters, right? So, how does disease affect brain drainage now that we have this fancy tool? So here's what we're going to do. We're going to, this poor mouse, we're going to inject LPS or PBS intraperitoneal, I can never say that word. We're going to inject towards the backside of the mouse, so we're not injecting directly into the brain, but in the body itself. LPS, which is our inflammatory membrane sugar, or salt water, this is our control. And we're also going to inject a fancy dye called cadaverin, named as such because it smells like a dying body. And we're going to then quantify ZSG leakage in the blood. So how does this affect leakage? So the thing you have to know about cadaverin is that it cannot enter vessels in a functioning blood-brain barrier. Right? So the thing to note about cadaverin is that it will only stain if the blood-brain barrier is compromised. So again, observe first, then give me your interpretation, right? Observe first, and you don't have to worry about this on the bottom, GFAP CD31. This is talking about other cells. I don't really care about that. It's probably important for the paper, but you know, it's getting late into the paper and I want to eat lunch. So these 4 panels is what we're looking at. PBS injection here, this is our control, our salt water. LPS here, cadaverin labeling here, and then ZSG here. So how does LPS affect blood-brain barrier permeability? Remember, cadaverin cannot enter the blood-brain barrier if it is functioning. It's too big, doesn't fit. So again, observation first, give me what your eyes tell you, and then give me the answer to the question, how does LPS affect blood-brain barrier permeability? In PBS, it is more diverse. What is more diverse? What is more diverse? What is more clumped? LPS equals blood-brain barrier is cooked. Salad flu. Give me the observation first, then the interpretation. I'm going to keep on reminding you guys this. Observation first, then interpretation. What is the green thingy? It's the neuron marker. It's the permeable green protein that the neurons are making to trace the drainage. Yeah, more green in LPS. That might not be entirely true. To my eye, they look the same. I think they're about the same number of neurons, if I were to guess. They look pretty even to me. More purple with LPS. Thank you, Salafu. And more purple in LPS. Now give me your interpretation. So there's more purple in the LPS injected mice. What does that mean for the blood-brain barrier? What does that mean for the blood-brain barrier? Cadaverin is very visible in LPS, but not that much in PBS. Yes, good observation. That is the language I am looking for. What does that mean for blood-brain barrier permeability now? Now you can tell me what this means. Now, what does this mean? It's cooked. Swiss cheese, leaky pipe, LPS makes blood-brain barrier not work. Yes, exactly. Yes. So the full length of the thought, right? Again, we're being very careful. We want to be better scientists. So we start with the observation. We never jump to the conclusion. We first describe it because that's how you can think clearly. That is how you avoid making mistakes. The data. Cadaverin labels something in LPS treated mice as opposed to nothing. The interpretation is the vessels are more permeable in LPS treated mice. Okay, that's how you walk through the interpretation. Prevent mistakes. Have clarity of thought. Okay, let's continue. So this makes sense. This is consistent with what we know about the blood-brain barrier. When you have a brain infection, the blood-brain barrier gets very leaky. The immune cells can rush in, deal a bunch of collateral damage. That's why blood and brain infections are so deadly. It's interesting to note that certain brain borders are leaking ZS green. I'm not going to spend too much time on this, but just so you know, this ZS green loss cannot be explained by an overall green loss in the brain in response to LPS. So people were saying it looks like there's less green in the LPS. Maybe, but it's not statistically significant, so we can't actually say that it is actually different. So, you know, the difference in these brain borders, we're seeing that in LPS, you get less of that green in the brain borders, which is interesting. When we test the blood, we actually see that the green protein that the neurons are making actually make their way into the blood when you inject with LPS. So not only is the blood-brain barrier leaky, neuron proteins are leaking out of the brain and into the blood. That's what this blue bar is telling us. And when you inject with PBS, you can actually see that it is a little different. I'm guessing because, but it's not statistically so, I don't think. But when you inject with just salt water, it's not really that different. But when you inject with a bacteria component, your brain gets leaky and then the neuron stuff ends up in your blood. Seems bad. Yeah, it does seem bad, doesn't it? So, what about amyloid beta? Now that we know that our dye works the way we think it works, because we already know that LPS does this, right? This is not new information. This is not new information. We're just checking to see whether or not LPS correctly predicts the blood-brain barrier permeability problem, and it does. So now that we know LPS confirms that this is working normally, that our labeling does in fact reflect physiology and disease states, what can we say about amyloid beta, which is our culprit for Alzheimer's disease? So here we have a genetically modified mouse that makes a lot of amyloid beta, which we believe contributes to Alzheimer's disease. And then after we do our AAV strategy again, we wait for 14 days, the brain lights up. Now, what are we seeing here? My question for you, chat, is, does our mouse successfully make Alzheimer's protein? In here, this is the column for Alzheimer's protein. This is our column for neurons making the dye. And then these are the overlaps. And then these are the zoom-ins. So chat, observation first, then conclusion. Do these mice successfully make amyloid beta protein? Do these mice successfully make amyloid beta protein? Give me the observation, then the conclusion. Observation, then conclusion. You're giving me the conclusions. Give me the observation. I'm going to make you do it. More red. There's nothing on top, a bit of red in the bottom. Yes. There is less staining of the amyloid beta in the wild type condition. Yes. Yes. Those are the observations. Again, training you to have clarity of thought. It seems easy now, but again, there were a couple of trick questions here where if you didn't do it, you would have been fooled. So even when the data look easy to interpret, observation first always, and then conclusion. Observation first, then conclusion. But I'm going to have to agree with you guys, because there is more red in this square. There's basically no red in this square. We can conclude that our Alzheimer's mice is in fact successfully making the amyloid beta, the Alzheimer's protein. Great. Perfect. All right. So what's happening in the Alzheimer's mice? How is brain drainage affected? So now we're seeing ZS green accumulation in the brain, right? And so there's no fancy picture anymore. Now we're looking at hard data, and now we're looking at the data itself. So again, observe, then interpret. What is happening in the Alzheimer's mice? And this one, it is really especially very important to observe first and then interpret, because I'm telling you right now, this is a trick question. So what's happening in the Alzheimer's mice given this data? First, give me the observation, and then give me the interpretation. First observation, then interpretation. And I'm telling you right now, this is a trick question. This is a trick question. What are you seeing? Tell me what you're seeing. Observation. Sal says there is significantly higher ZS green intensity in the Alzheimer's mice. Yes, that is exactly the language of observation, because the interpretation is going to be very, very tricky. Okay. It's going to be very tricky. Okay, so what is the interpretation then? How is brain drainage affected in this data? What is happening? I'm telling you this is a trick. I'm telling you right now this is a trick. From this data, how is the brain drainage affected? Who wants to say? How is the brain drainage affected? This is a trick question. Trick question. It's stuck inside. The modified mouse has less drainage. Drainage doesn't work in Alzheimer's mice. I said this was a trick. I said this was a trick. And you guys fell for it. From this data, if you accept that the drainage is what is the issue here, you just, as my advisor would put it, drank the author's Kool-Aid. You uncritically agreed with the authors because of the framing. I asked you, how was the brain drainage affected? I led you to a conclusion, but I told you it was a trick for a reason, right? This is true skepticism. For all the people who say, how do we teach critical thinking? This is critical thinking. Okay, this is critical thinking. It is the ability to criticize what I am doing here, how I am manipulating your perception of the data, which is the author is going to do to you because the author wants you to agree with them. But from just this data, can we say that the brain drainage is affected or not? Some of you have correctly suggested, no, we can't. Why? The observation is that these Alzheimer's mice have more ZS green. That's great. That's a great observation to make. The interpretation is either the synthesis of ZS green is greater for some reason, maybe the neurons are just making more of it, or these mice have a drainage problem. Or these mice have a drainage problem. The authors want you to believe that it is a drainage problem, but until we see evidence of that, we should remain skeptical. That's how you improve your thinking. That's how you have clarity of thought. That is why I always ask you, observation first, then hypothesis. I'm sorry if I sound mad. I'm not mad at you. I have no incentive as to whether or not you learn or not. I'm just very passionate about this, right? Like, I've been speaking with teachers, and there's a real lack of critical thinking in students, even at an older age. And I think it's because it's hard to do this kind of learning, right? I'm grilling you. I'm making you feel uncomfortable. I'm telling you you're wrong. But that is the level of rigor that's needed for proper critical scientific thinking, right? I'm not mad at you. I'm not disappointed because I set this trap up for you. I want you to remember this forever. I want you to remember the feeling of being bamboozled so that you are not bamboozled again in the future. I bamboozled you in a safe place. You're allowed to be bamboozled here. Bamboozle is a part of the learning process. And then when you get to your labs, even in your day-to-day life, when you read news online, when you read a paper from Reddit or whatever in your life requires you to skeptically think, Observation first, then conclusion. Otherwise, you will be bamboozled. Okay, you will be bamboozled. It's easy to be bamboozled. I just did it to a lot of people here. So yes, it could be that it is more is being made, could be a drainage problem. But until you have more information, you cannot conclude either way. And I don't really think that they do that in the paper, which is disappointing. So it's an asterisk, right? But they have plausible mechanisms to do so, right? They label amyloid beta, and amyloid beta appears to accumulate at these sites of drainage at these neurons. So it's possible that they are blocking up the cell, but we're not sure. This is correlative, right? Amyloid beta, these are regular exit points that the neurons would normally be shoving things out, and amyloid beta happens to be clogging these exit points out. This is a nice correlation. It is consistent with the idea of drainage being affected, but I did not show data that confirmed it. Correlation isn't causation yet again. Yes. And you will be fooled by this repeatedly until it gets drilled into your head, right? And if you keep on attending these journal clubs, you will learn very, very well the difference between correlation and causation. You will learn it in your soul, because everybody on Reddit, everybody in R Science wants to believe that they know correlation versus causation, but a lot of them are also being fooled as well, right? When I stream, that is when you actually learn. This is one of the locations when you can actually learn how to critically think. I'm happy that all 100 plus of you here today are doing that. It makes me very happy. It makes me feel like my work, even though I don't get paid to do streaming, right? Like I make a lot more money in ad revenue. I still do these streams because I believe so strongly in this mission of encouraging critical thought. Okay. So even though they didn't rule out synthesis, I will say this, right? I don't want to be overly critical. I don't think there's a good reason to believe that there is more production of this green dye in Alzheimer's mice, right? I'm not saying that this paper is wrong. I'm just saying it would have been a little better if we had proof that they weren't somehow, for some unknown reason, making more of that protein. Like, it's possible. It's not likely, but it is possible. I will still interpret it as a drainage problem, but I'm a little more careful with that interpretation, right? I'm a little less confident. And what I'm trying to illustrate for you, I'm trying to model for you, is how I talk about my understanding of the data. It's not I believe versus I do not believe. It is I am confident, I am less confident, I am not confident, I'm a little confident, right? And because they didn't rule out a difference, or at least maybe they did and I just missed it. I don't want to crap on the authors. If they did, I'm sorry, I just didn't see it. Because I didn't see them ruling out more protein synthesis as being an option, I am a little skeptical of brain drainage being a thing. Not a lot skeptical, a little skeptical. Just a little bit. Just that nuance. Okay, keep in mind. That doesn't mean I don't believe the authors. It just means I don't believe them 100%, which is fine. I think the authors of this paper will be very happy that I am even 85% on their side, because that is how you should think about the truth. Not yes or no, not true or false, not black or white, but gray. Dark gray, light gray, okay? Think carefully. So this is kind of their summary of the disease states here. LPS allows drainage of proteins into the blood, and then amyloid beta seems to be doing the opposite, right? In the brain, we saw that there's an accumulation of ZS green, whether or not it is because of improved synthesis, because of some weird glitchy effect, we don't know, but it's not likely to be because of synthesis. And then it decreases in the borders, and then it also decreases in the blood. I didn't show this because I'm tired. But yeah, later they show that like Alzheimer's protein, unlike LPS, which makes the blood-brain barrier more leaky, a lot of those neuron proteins end up in the blood. When you have the Alzheimer's mice, there's less neuron protein in the blood, indicating potentially that the neuron proteins are kept inside the brain. Yeah, it is possible in Alzheimer's, the epigenome is deregulated. Almost all genes deregulated. Exactly. That's why I'm like, it's not like super likely that it ramps up this random protein by a lot, but it is possible. It is possible. Okay, big summary. So genetic labeling might be better than injection labeling because you can see more things that are likely physiological because you're not pushing a liquid through the brain and affecting inflammation, etc., etc. You can trace drainage with these genetic systems, right? You can even see how fast different regions of the brain drain. And we can see that immune cells are doing stuff in response to this label. We don't have raw evidence that they're doing stuff, but we have light evidence that they're doing stuff from their genetic regulation. ZSG drains locally as opposed to one big circuit. And when LPS inflammation is present, the brain gets real leaky. That is consistent with what we know about LPS. That's not new information, but it's consistent. So that means the label is working the same way as other labels that we know of so far in this particular use case. And when amyloid beta is present, the brain appears, okay, I'm being careful here because again, they didn't show, or I didn't show that protein production isn't affected, but it appears that the brain seizes up and gets backed up with additional neuron protein. So nuances and caveats, right? Mice don't get Alzheimer's disease, of course. This is the number one thing that we all have to keep aware of. That doesn't mean we throw out research that has to do with mice and Alzheimer's disease. Because, oh my God, it makes me so frustrated. People love to simplify stuff online, right? Whenever you see an Alzheimer's mice paper on Reddit, there's some wise Alec in the comments going, well, mice don't get Alzheimer's disease. Yes, that is true. The next sentence has to be, and so we must interpret the findings with caution. Instead of, that means the study is bunk, which is a lot of what Reddit does. Mice can be very useful. They can provide novel insight into human biology. You have to be careful that you don't equate the two. You have to always then see how you can make sure it actually happens in a human. To give them Alzheimer's in this particular mice, 5 entire copies of the human amyloid beta gene are genetically engineered, right? So this is a very artificial system, right? Because Alzheimer's is pretty rare in humans too. There's not one Alzheimer's gene that gets mutated and then you have Alzheimer's. It is polygenic. There's a lot that can go wrong. There's a lot of different things that can go wrong that all converge on the Alzheimer's. So it's a pretty artificial mouse. So mice aren't people. 5X, I think familial Alzheimer, amyloid beta. That's not how humans get amyloid, right? This is a very artificial system. However, this shows that in a mouse model, excess amyloid beta is sufficient to clog brain drainage. So I'm not claiming anything about Alzheimer's disease in this paper, right? Not claiming anything about Alzheimer's disease. But what we are saying is that when there is amyloid beta present, it clogs the brain. Asterisk. Pretty sure it clogs the brain. 85% sure it clogs the brain. Why can't we use alternate animal models? I always wanted to use monitor lizards. Mice are cheap. They breed very quickly. They have an entire genetic toolkit that makes them very easy to manipulate. Every other animal is likely going to be a lot more expensive, besides like flies or worms and stuff, right? One great ape, how much do you think one ape is? Lifetime cost. To have one simian, just one monkey. Guess, chat. It's a good question, Ace. One ape. Just one. Lifetime cost for one. Some good guesses. Millions of dollars, millions of USD for one single primate. Fungible ape. Yeah, true. Well, then it's very inconsistent, right? Depending on whether or not you're going to the moon. Why is it millions? Well, you have to care for the entire, you have to upkeep the zoo, the enclosure that they're kept in. You have to pay all the vet techs that keep the ape happy and enriched, right? They have to have a lot of enrichment. They're complex animals. You have to do a lot for them. Otherwise, they get very upset and sad. Research animals, if you're a research simian, depending on what experiment you're in, you actually live a pretty blessed existence because a happy research animal is one that gives you good data. So there's an incentive to make sure that these animals are treated well. And it is also a reason why we don't use animals for no good reason, right? We only go to a monkey or an ape if we have a really, really good reason. Like, we're not just throwing apes into these research labs without a really freaking good reason to. And a mouse is very cheap. A mouse is, back when I was in graduate school, inflation and not, but when I was in graduate school about a decade ago, it was about $1 per mouse per cage. And they don't live that long. They live 2 years. So instead of millions of dollars, it's more like hundreds of dollars, much cheaper. They breed much faster. So, lizards, birds, sure, you can, but is it appealing? Is it attractive? Probably not. Mice are just so much better. They're fast. Their genomes are very easy to manipulate. They are good enough at looking like a human, right? If you want to talk about monitor lizards, well, we can't really learn a lot about our biology from looking at a lizard. You can't learn nothing, right? In my next video about regeneration, I'm going to be talking about axolotls, which are useful for studying regeneration, but mice just gives you so much more. So much more. It's crazy. Okay. So, again, we live on nuance on the Fi the Neutrophil channel. I will, as best I can, try to give you the full nuance as best as I understand it. Nuances and caveats, I would not take away that much about Alzheimer's disease here. But I will say that I am confident that Alzheimer's protein is at least affecting brain drainage somehow from this paper. That's about as much as I would take away. So in an Alzheimer's patient, I would guess that the presence of Alzheimer's protein, amyloid beta, I would guess that it is affecting brain drainage. Whether or not that turns into dementia, that's a different question. I don't know. Not from this paper, at least. But that is kind of my nuanced understanding of the paper. And I don't think the authors would disagree too much. At the very least, we have a new tool, a new mouse. New mouse just dropped. All right, this mouse took a lot of effort, right? This is a cell paper. Any new mouse model is going to require half a decade of work and millions of dollars to make. So at the very least, we have a new cool mouse that allows you to label the brain in what appears to be in more detail. Although future experiments will have to be done to really make sure that this is actually representative of how we know the brain to work. But I think it's still pretty cool. I think it's still pretty cool. So, questions. We are at the end of the journal club. Thank you all for making it to the end here. It's not an easy one. It's a very tough one. But yeah. What do you guys think? Oh, it's $1 per mice per cage to upkeep them. To buy a mouse is a couple of $100s in itself. Sometimes a couple of $1,000 depending on how rare the mouse is. But even so, like a couple $1,000 compared to a couple million for a simian is a whole nother level, right? Whole nother level. Mice are great research animals. Fantastic. If you don't like the idea of researching on mammals, though, if you have, concerns about animal welfare, that's totally valid too. If you want to do biology, then you can choose to do it in simpler animals, flies, worms, zebrafish, or even bacteria. Tons of bacteria researchers out there too. If you don't want to work with mammals, it's also fine. Any questions? Any general updates? Any general things you guys want to chat about biology? Now we can enter the just chatting portion of the stream. So thank you all so much for coming today. Now I just want to reconnect with my audience because I've been busy. I thought, when I first got monetized that like maybe I would stream a journal club stream every week. But that turned out not to be plausible because again, the streams don't really make money. Gotta make the money first. But I love doing these and I'm very happy and just happy to be with you guys, So what's going on? What's going on? How's school going? How's learning going? That's often what I like to talk about because I am so invested in the lives of students, right? Even though I don't teach anymore because I got let go from my job, I love hearing about like how learning is going because it's just interesting to me. And you can ask me questions about like biology or being a teacher or anything like that. I'll probably chat for 15 minutes and then I'll call it a stream and then I'll go have lunch. What am I having for lunch? I am having Boston baked beans and some roasted potatoes that I made last night. Yeah. Does the immune system do a tolerance increase to protein only if it's made inside the body? If it's made by a cell, it, naturally, that is one avenue by which, the immune system can learn to tolerate cells. Primarily, T and B cells will learn because there is a cell called the MTEC, and it shows the T cell and B cell, hey, this is something that we made, not something that a virus made. You should learn, or rather, if you react to this, I will kill you. If you don't react to it, you can survive. So that's the major way that tolerance is established. There are more complicated methods as well, stuff that I don't even fully understand myself, but I will say that as a simplification. I have a question. People say animal models are useless because a lot of animal experiments don't apply to humans. Are they right? This is a thing about the thing about nuance, all right? Yes, mice are not humans, but mice have a lot of our same biology, and that makes them still useful. Thank you so much, Brady, for the gifted subs. Thank you so much. Everybody make sure to say thank you to Brady. Thank you so much. That's awesome. Thank you, thank you. Make sure you go enjoy the emotes and the non-science content that I have as bonuses here. But thank you so much, Brady, for the 10 gifted. Huge. Absolutely huge. So they're not right. Animal experiments are absolutely necessary if you want to understand anything about a human, unless you want to go right to monkeys, which is a far worse crisis for animal ethics, right? A monkey is pretty similar to a human. That is definitely true, but we're talking about a lot of suffering of a much more intelligent animal. I'm not here to play scorekeeping with what animals we consider ethical and what animals we don't. This is just how the system works, and I'm describing that to you. So unless you want to rear monkeys, you just can't. Unless you want to subject humans to experimentation where we don't know what the consequences are, you have to do it in an animal, right? An organ system that you do in a chip can also be very powerful, right? There's this huge push for in vitro experimental models like organ on a chip, which are great, fantastic, love that. Organs don't have immune systems though. Right? You will never capture the full complexity of biology in a dish. It's just not possible. You can do a lot, don't get me wrong. Those are still amazing experiments, but they will never replace having an intact animal, because an intact animal has organ systems talking to each other. So no, I hate that argument. I see it all the time on Reddit. It's in an animal and therefore it can't apply to humans. Yes, but let's talk about nuance. It doesn't mean it's not useless. It just means it's not 100% applicable to humanity. But if it's 85% applicable to humans, I bet you wish that experiment happened in an animal than in a human. Sorry, another soapbox. Does bioinformatics a good degree to get? I'm still in high school. I think bioinformatics is great. A lot of it is going down the route of AI, as all programming-based stuff is, but big data is the name of the game, and it depends on how safe you think your jobs will be after AI, because there is such a AI push in all regions now. But I will say it is very valuable. Bioinformatics is a very valuable, field, but only, I'm not a bioinformatician, so I can't tell you about the job market for that kind of stuff. You should talk to somebody who actually is in the job market, and they can give you a more accurate reading as to whether or not that's a good field to go into. But I will say it is a valuable field, but maybe not for a researcher because of AI. Again, AI disaster. Kind of a disaster for a lot of things. Zebrafish, that's me. Nice. Love zebrafish. Read an article on Alzheimer's vaccine and most of them failed. I mean, most of medicine fails. If you only knew how many millions and trillions of dollars go into medicines that don't work, I'm not surprised. It's very hard to make medication. What is this squiggle in your eye? DNA? Yeah, it's my chromosomes. Starting my second year of college soon, I suppose. That's nice. Getting to start your second year, that's such a nice place to be, because I feel like freshman year for me was very awkward. I spent a lot of it trying to make friends and failing. But second year, I start to feel more comfortable, not only socially, but just in my own being. So that's awesome. I love college so much. How do you know that color isn't just a visual thing? Electric perceive electric signals that we perceive color. I mean, sure. I'm not entirely sure where that question is going. All color is a specific reaction to a specific wavelength. So I'm not a firm believer in color being a thing. It's just a representation of what wavelengths are underlying a particular phenomenon. You're a biology teacher? I used to be a biology teacher. I got let go a year ago. A year ago and 2 months. And since then, I've been a full-time YouTuber. Yeah. I know nothing about biology and failed chemistry twice, but I love your content and learning about biology. I'm glad. I also love biology. And I also had not so good grades in chemistry, so I do relate to that. Why'd you get let go? AI made it so that my students stopped caring about biology and cared more about AI. I had one semester where I legitimately had a class of 3. And, I don't blame my university. I probably would have let me go too. And then, general university funding in America, if you can remember what happened a year or 2 ago, not so good, So I very much sympathize with my university's choice to let some quote unquote dead weight go because of lack of funding and also students being more interested in AI than in biology. But now I get to talk to 100 of you on stream and hundreds of thousands of more on my videos about biology. So eat that, AI. Did I do a PhD in immunology? I did not. I did not finish my PhD. My PhD was in neurobiology, neuroimmunology specifically. Yeah. I'm scrolling past the gifted subs. Thank you again, Brady. I'm doing my capstone on delivering genes to stem cells. I'm using some AAV construct, but it's a bit more boring than I thought. It's about what you make out of it. Things are only as interesting as the stories you tell yourself. I'm sure you can find a way to make it more interesting. I'm not in school or academia anymore, and even though I do work with science, it's mostly repetitive chores with little space to learn new things. That's why I do these journal clubs, right? If you think about it, I'm learning for my job, which is YouTube, but to get really deep into a paper, I can't do that for a video. It would take too long. It already took me a couple of hours, not only to do the presentation, to teach myself the paper, but also to do this, which is to actually teach. And when I'm actually teaching, that's when I'm actually learning. Do mice genuinely not get Alzheimer's, or do we not interact with them in ways where we can't see its symptoms? They just don't. I don't remember if they don't make amyloid beta. I'm not sure if they have a natural amyloid beta gene. I seem to remember that they don't actually make amyloid beta. That's more of like a simian thing. And also they live for like 2 years. So even if they did make amyloid beta aggregation is a disease of old people, and mice just don't really live that long for protein buildup to occur. Do vets also gain from animal studies? So vets have to be employed. You have to make sure your animals are happy and healthy if you want to do research with them. So I'm guessing animal models help us to guess things about how humans might work. Exactly right. What are your thoughts on simulated biology? Is it possible to do a useful study of biology in a computer simulation? I think it is possible to do things that are mildly useful, I'll put it that way. And a lot of universities that don't have access to incredible technology have to rely on computer simulation for certain hypothesis generation, which I think is fine. A lot of protein modeling stuff is done on the computer, but I think for me as a bread and butter, brick and mortar molecular biologist, nothing beats a Western blot. Nothing beats like hard evidence for me as someone who is of a different age, because now we're all doing computer stuff. Yeah. Speaking of brain chips, I heard a lab brain reached criticality. What does that mean? Don't know. And I am not willing to entertain it until many years into the future when I can fully understand the nuances as opposed to reacting to recent news. I'm not saying that to be snarky. This is a general policy on my channel. If you are asking me, oh, did you hear about this newest breakthrough? Chances are no. I don't actually keep up with the newest, greatest, and latest breakthroughs until I am confident that they actually mattered. If there's a whole field that, blossoms out of it, if it gets cited repeatedly, if it gets proven to be true and impactful, then I will care about it. But if the question, generally the question of, did you hear about X and this thing came out in the last year? Probably no, because I like to wait until the whole story unveils itself. Yeah. Reddit bros casually forgetting all of our DNA is interconnected and intersected in some way, shape, or form across all species. Exactly. We all come from the same primordial soup, you know? We all crawled out of the primordial soup. We all have a shared lineage, a shared lineage. All of our cell cycle genes are shared to a high degree. They're very well conserved. We have a lot of overlapping biology. It's just some animals are more similar to us than others. Yeah. Where are you in my studies? I dropped my PhD. I've been out of graduate school for like half a decade. Why do both microscopes and people see green better? Are microscopes and humans more genetically related than mice and humans? That's pretty funny. In terms of taking the question seriously, I don't know if green is necessarily the best for human visibility, but for microscopes it has to do with quenching and excitation and bleaching and all this other microscope person talk that I'm not qualified to talk about. Microscope people go hard though, in terms of getting to, you know, if you find a microscope person, they could probably tell you why. I mostly come from a maths background, but your stuff makes me think biology is the coolest. I'm glad I could be the bard of biology, as I like to repeatedly call myself. I'm like a bard. I sing the stories, the best ofs, the greatest hits, because I love this field so much. And I'm glad to be able to, you know, take revenge on AI for taking my job, right? To hopefully encourage people back into biology. Yeah. Yeah. TBH are a great teacher. Bet they are regretting their decision. I worked at one of the best universities in the country with some of the best faculty in the country. I'm sure they actually don't miss me at all. I'm not saying that to be self-deprecating. I appreciate those kind words, but I like to be realistic with myself. I can do a lot more on YouTube than I can do in that classroom. I think I've been reflecting on it. Like, I was the youngest member of the faculty, and so I wasn't given a lot of independence. I was just given the script, and I couldn't do a lot of my own creative expression of teaching. And until those other people I worked with retired, there's no way that I would be able to do it on my own. And I didn't have a PhD, so there's no way they could let me, quote unquote, spread my wings and fly. I was always going to be the worker and not the leader in that environment. So I don't think they regret it. They let go of someone without a PhD. They probably don't feel too bad about it. But I'm more of an artist and a communicator than I am a teacher. Even though I do really, really love teaching, I am more earnestly a communicator than I am a teacher. And I do believe there is a difference. I'm not even aiming for university, but I'm glad you go to your classes. That's very kind of you. You can continue to show up to these journal clubs, again, about once a month. I want to do computational neuroscience, but I did a coding internship this summer, and now I really don't enjoy vibe coding, so I don't know what to do now. Yeah, I unfortunately don't have a lot of insight for how to interact with AI in this day and age, because it is sad what's happening with the most exciting stuff about science being outsourced to AI. AI can do some great stuff like crunching through protein folding. The problem is it's a new hot thing, so way too over pushed right now. Yeah, exactly. Dragons. It's really overhyped because we have machine learning, which is not quite the same as LLMs. And machine learning has been a part of neuroscience for a long time, but the problem with vibe coding is that is LLMs, and I think LLMs are, in the majority of applications, not good for humanity, but selectively good for humanity, right? I'm not saying they're bad all the time, but for certain research applications, they're great. For others, it just makes everything terrible. Talking about Alzheimer's, you know that the completely new human organelle hemifusosome found last year? I have heard of it. Acting as loading docks where tiny cellular sacs connect and move cargo. I have heard of it. It is on the list of weird organelles, and I'm planning to make a whole video on weird organelles, so it's definitely on my list of things to talk about. The real reason of Alzheimer's is not known. Exceptionally familiar Alzheimer's is due to mutations of some genes of the amyloid precursor pathway. Alzheimer's only exists in AD. Today's AD studies on AD brains or IPCs. It's complicated, yeah. What's more similar to humans pigs or mice? I've seen both. Pigs have more similar organs to us. Mice are just good generally, and again, for all the reasons that they're cheap, good at reproducing, fast, etc. Yeah. And alongside biology, any other sciences you're interested in? I love psychology, and even though this isn't really a science, but I love philosophy as well. I wanted to be a psychologist at one point. I love how messy psychology can be, but it's almost too messy for me. And that is not okay. Soapbox, regular psychology defender soapbox. Okay. Molecular biology has very robust tools, but physicists would consider molecular biology too messy. And a lot of biologists would consider psychology too messy, and I agree for my personal taste. However, I believe psychology is valuable. I believe that the replicability crisis first hit psychology, and they are doing the most to attack the replicability problem. Because guess what? Psychology isn't the only isn't the only field with problems in statistics, right? So before I get any psychology is hard, I get it every stream. I might seem like I'm having an overreaction, but every stream there's some wise guy in chat that goes, psychology isn't a real science. It is. They just happen to take the bad PR for the replicability crisis, and they are the frontline vanguards, the frontline warriors on how to make all of science better. So shut up with psychology isn't real or less good. No, I will insta-ban you for that. I am so tired of those wise guys. Psychology isn't... Have you seen the statistics in a psychology paper? Like here, like in biology, like you get a very minimal statistical education. In psychology, they like teach you like arcane statistical arts that like most scientists would never even know about. Okay, their understanding of statistics is much better than the average scientist because they were the ones who reckoned with the replicability crisis first. Okay, I love psychology. Shut up. Shut up. And I also love philosophy because that's more of the art side of me. Thinking about the human condition is a very artsy thing to do, and I love doing it. Human condition, right? Biology is a part of the human condition. Psychology is a part of the human condition. Philosophy is a part of the human condition. You are our teacher now. Thank you, Phi. Thank you for all you've done. You're welcome. And thank you for showing up and just providing the support I need to feel like the work is worthwhile doing. I can't wait for the LLM bubble to crash in the US. It's already crashed in South Korea. I sure hope so. As a primarily chemistry and physics person, seeing all the normally lifeless atoms in the form of funky proteins chugging away is so cool. I love molecular machines. I love molecular machines. Clockwork. Oh man! Eventually, I hope Clockwork does a video on kinesin and dynein and he's already done one on myosin, which are the motors for muscles. But I love the fact that there is a lifelike element to molecules. It's amazing where we are built of tiny life. Watched your dark proteome video onto my oncomaterials lab I work in. MHC1 is also my favorite protein. Sick. Love MHC. Fantastic protein. Philosophy, yes. Maybe I'll do a philosophy streams too. I love philosophy. Messy like the footballer, true. I do a bunch of research on neuroscience stuff, and this channel is very cool. Thank you. I come from a neurobiology background, so I'm glad to hear it. We have 2 psychologists and 1 psychiatrist in VTheory, by the way. Exactly. Let me go ahead. Oh, I don't think I've updated. Did I update this command? Did I update the command? We have 2 psychologists. We have Myelin and Sidere for psychologists, and we also have now Lev Velikor, a psychiatrist. So the fusion between psychology and biology and medicine. Unfortunately, I don't think I updated my Stream Elements bot to reflect her channel, but you can find her, Lev Velikor. I'll shout her out here. If you want to check her out, you can just Google her name. Our resident psychiatrist. Is being a YouTuber sustainable for you? So I am making a paycheck now. It is more than my state's minimum wage. So I'll just say that, right? You can come up with your own conclusions, but I can proudly call this a job, right? I'm making more than minimum wage. It's no longer a hobby. This is my full-time job. Whether or not it pays a lot, well, yeah. Patreon, YouTube membership, buy my merch, blah, 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 all those other plugs. But I am able to make a living, a minimum wage living off of this channel, and therefore I will try to hopefully, build up the channel. It's still young. I've only been 1 year out of a job at my former university. And already in that 1 short year, I have turned it into a minimum wage job, which is sick. And, hopefully it'll get better in the future. Did I see the new study about octopi? Again, with new studies, I tend not to look at new studies. Also, I'm sorry, I'm not really a touch grass kind of biologist, so I will usually not read stuff about more complex animals. The most complex animal I will normally do reading about is a mouse, and if we're talking about human studies, then humans, right? Otherwise, not really. Do I have merch? Yes, you can find it on my homepage. And just like if you just scroll down, oh man, it doesn't show. If you go to my YouTube channel, there's a tab that says store, and then you can buy merch there. There's some new merch dropping soon, by the way. So if you're gonna buy merch, maybe you wait to see what else I have cooking, and then you can buy merch then, because I think I have some great designs that I think are pretty interesting. How did you learn to draw like this? It's so cool. I do the bad drawings on my channel. If a drawing looks good, it's probably because Toxo drew it, my wife Toxo, who supports the channel by making the thumbnail art, by making a lot of the assets, and then the crappy drawings are the ones that I make. Yeah. I was making devlogs for a mod I was making. I didn't know the subscribers needed for enough money to get a paycheck is so many. It seems like a very intense challenge. It's different for different niches. I'll say for education, it's like middle of the pack in terms of earnings. And also, it's more about how many people watch your content versus subscriber count. Subscriber doesn't really mean a lot on YouTube anymore, but how many views do your videos get? That's why I always plug my membership at the end of videos, just to be like, you know, YouTube is up and down, but like membership and Patreon, those really help give me a stable core of income that doesn't change month to month. But I wouldn't worry, like I'm not starving, right? Exceeding minimum wage is like very, very nice, and I have savings. So I'm not like, you know, begging for money. It's just the reality of the question of can you make a living is yes, but it's complicated is basically what I mean to say with that. Yeah. Biophysicist here. Biology blows my mind every day. NK cells are underrated. They will be getting their spotlight at some point. I do love NK cells. I love the role that NK cells play in the story of immunology. So I think in the next year, I might make an hour-long video either on the history of the immune system or on the regeneration of a single cell to a whole embryo. You know, the story of you, zygote to full human. That's also like an insane topic, but like the story that NK cell plays in the story of immunology is totally crazy. Love that cell. It's so good. Fi is married, hell yeah, hell yeah. Toxo emote. Thank you, Seed. Did you see that YouTube is no longer counting views but clicks now? Ah, it's terrible. I'm not going to get too much into it, but I don't foresee that being a smart decision. Just hope I can weather the storm. You into embryology? I've been fixated on that for a while. My next video was on regeneration, which is essentially a subset of the study of developmental biology. Would I recommend neurology or neuroscience? Neuroscience if you love programming, neurology if you kind of like programming. I like neurology. I think it's a little less prone to being outsourced to AI, you know, with all the vibe coding and all that. But depends on the question that you like. Neurology is about the biology of the brain, whereas neuroscience is about the connectivity, the signaling, how you go from patterns to thoughts. So it just depends. One is not better than the other. It depends on what questions you like to ask, and it depends on what AI is going to do to these jobs in the future. What's the difference between views and clicks? Views require you to actually stay on my video for a little bit of time, whereas clicks are just that, clicks. All right, I am getting hungry, but thank you all so much for attending this journal club. I love doing these. It was so much fun, and I can't wait to do the next one next month. Don't know what we're doing yet, but if you are macrophage or above, you get to vote on which paper I cover. I'm sorry, I can't really offer that many rewards because my life as a YouTuber is so busy, but I did want to give macrophage and above one additional perk, and that is basically picking the journal club. So thank you all so much for the support on the channel. Even if you don't monetarily contribute, I'm just happy seeing the number of concurrent viewers. It just means a lot that I get to help people understand science in a way that I don't think a lot of people are doing online. And it's valuable to me to know that there are this many people of you watching. So thank you so much for being here. And I will see you later. Yeah. Have a great day. Bye, y'all. Bye-bye. Bye.